Every year, 29th April is celebrated as International Day of Immunology, with an objective to increase the global awareness about infection, autoimmunity, and immunology. And recent years, like since last 1 year, immunology has definitely picked up as a interesting topic for everyone because of this COVID pandemic, and it does not require much introduction as it required few years back. So this, to mark this day, International Union of Immunological Societies and Indian Immunology Society has floated the theme of COVID-19 for this year, considering the pandemic. So Department of Microbiology, in association with Indian Society for Immunology, is having an online event today where in the first half we will have few expert lectures, followed by in the second half we will have an expert lecture and followed by some of the activities by student participants. First of all, I request Professor Vusudika to please welcome the guests.
So, good morning, everyone, to the event organized by the Department of Microbiology for the Immunology Day. I would like to first invite all the speakers, our distinguished speakers. So, this is this event we are organizing under the leadership of our Vice Chancellor, Professor Tiwari. So, today, what better topic will be there than COVID-19 related immunology. So that is where we are going. So we have Dr. Subhash Varma, Dr. Krishnamurthy, and Professor Rama from our own institution. So they will give you a number of insights regarding the disease and how for the surveillance, infection, what are the immunological markers from a different perspectives, and all the students and all the participants here will immensely benefit from this program. So we have divided this into two sessions. So this is the morning session, and one we will have in the afternoon, where we have our guest of honor, Professor V.S. Chauhan. So with this, I will hand over to Dr. Monisha.
We have Honorable Vice Chancellor Sir with us. So before we start, I request Sir to give few words of encouragement to our team.
Good morning, everyone, and welcome to this very important, though brief program organized by Professor Monisha Dhiman of Microbiology Department. As already informed, we have three distinguished scientists who will give their opinion and current status of knowledge on various aspects of COVID and corona pandemic. Professor Rama, who has been a distinguished scientist and associated with our university for nearly a decade. Then we have Professor Subhash Varma. He is a distinguished scientist in Center for Molecular Medicine, University of Noida. Then we have Professor Dr. S. Krishnamurthy. So all these three, they have spent a lot of time, energy, and efforts in the researching in the area of immunology, and they'll share the series very rich experience with our faculty members and also with the research scholars. And I'm sure that the deliberations that we will be having today and some events in the afternoon, as we all know that students and research scholars, they should not be made only part of the audience. They in every academic activity that we organize, either in academic or research institutions, research scholars and students, they should be party to it, and they should be the stakeholder. They are stakeholders, so there should be platform, there should be occasion for them to ventilate their views and the talent and the skill that they have. And in this way only we can ensure that we develop skills in our students, and also we try to make them entrepreneurial and also give them chance to decide the future course of action that they like to have for their career. So that is why I'm very happy to see that the students they'll get a lot of opportunity to express whatever experience they have gained over a year and a half, and also what they would like to do in future. And this is very important activity, and I request the two scientists sitting here, the three rather, Professor Varma, Professor Varma, and Professor Krishnamurthy, to be privy to the students' presentation in the afternoon. Though you will be very busy, but if you see them what they want to say, it would be great encouragement for them. So with this, I again welcome and thank both Professor Varma, Dr. Varma and Dr. Krishnamurthy for spending their time and energy with us, and for deciding to inspire and guide our students and research scholars. Now you will also know about our university, what we do here, what our scientists, our teachers, what type of research they do, and also it will be an opportunity for our teachers to interact with both of you, of course, with Professor Ramarao. They have been interacting for more than a decade, so it will be really a chance for them, and this interaction, I'm hopeful that will lead to further collaboration and joint efforts in the area of research. With this, I thank you all, and I thank Dr. Monisa for organizing this beautiful event. Thank you. Jai Hind, Jai Bharat.
Thank you very much, sir. So now I hand over the next sessions would be coordinated and moderated by Dr. Somesh, Dr. Nasir, and Dr. Pramod. So now, Doctor, over to you, Dr. Somesh.
Okay, so good morning and welcome to the morning session of this International Immunology Day, a virtual event organized by Department of Microbiology of Central University of Punjab, and under the leadership of Professor and Vice Chancellor, Honorable Vice Chancellor, Professor R.K. Tiwari. So in this morning session, we have three important talk actually. So let me first introduce our first speaker, who is joining us live from University of Nevada, Reno, so in United States of America. And so thank you, Professor Suhas, for spending time with us, especially in this late hours. Actually, it's 9 p.m. at your place. So let me first introduce a little bit about Professor Varma. So Professor Varma is presently working as an associate professor in Department of Microbiology and Immunology, and he is also part of the Center for Molecular Medicine and at University of Nevada, and he is a trained virologist and molecular biologist. Actually, in my opinion, he is an accomplished scientist with more than 20 years of experience. So he received his PhD from Banaras Hindu University in 2002, and then he moved back to United States of America, and he did his postdoctoral training from University of Pennsylvania, where I met him actually. So this was a long time ago, I think 14-15 years ago I met him. So presently his laboratory is interested in understanding the biology of two viruses actually, moving from last 14-15 years ago. That one is this Kaposi sarcoma associated herpes virus and the Zika virus. But what happened during this COVID pandemic, using genome sequencing, his group was one of the first group actually in the United States who basically reported the reinfection of coronavirus in the United States. And they published this work in Lancet Infectious Disease also. And moreover, he is serving as a director of biomedical graduate program. So those who want to go for higher study, actually, so he's the right person. So I request you to please contact if you want to do PhD under his, if you want to join this institute. So the title of his talk is basically, today he will discuss this SARS coronavirus 2 variant surveillance and the case of the reinfection. So over to you, Professor Varma, and you may proceed with your talk. Thank you very much.
Okay, let me start to share. How do I go to share here? Presenter view, entire screen. And tell everybody see my screen now or no?
Not yet.
Not yet, sir.
It is it? Okay.
No, sir.
Not yet.
No? Okay, let me, okay. Can't, my no screen recording permissions dismiss. How about now?
No, sir.
No, sir.
Oh. How do I, a window maybe? Let's see, PowerPoint. Give me a second, yes. We should have figured this out before, I guess.
You can email us if you want. You can email us and we can upgrade from here. So you're mute. I guess we can see now. It's starting now.
Okay, do you see my screen now?
Should be there.
इसको pin कर दो.
Is it visible?
Yeah,
Yes, sir, it will be better.
okay.
It is? Okay.
Dr. Siva can see.
We cannot see.
You can't see? Huh.
Okay, it is visible to others.
Entire screen, let me see now this way. Do you see it now?
No, sir. Yes.
Yeah, now we can
Yes, we can. Yes.
Okay, all right. Sorry about that. We should have figured this out before starting. So, yeah, thank you, everyone. I don't see anybody. It seems like I was talking to the screen. Anyway, but we have to deal with it. So, thank you, Somesh, for a nice introduction. Yes, I know you from 2004, 2005, like 15 years ago, when we were at the University of Pennsylvania, and that's where I did my postdoc. And I was working at a virus. Our lab was a virology lab. We worked with DNA virus, and after I moved here at University of Nevada, Reno, I started working with RNA virus. And when this pandemic hit, then we switched to SARS-CoV-2 like many other laboratories. So the title of my talk is like SARS-CoV-2 variant surveillance and some cases of reinfection. So before I go and talk about those studies, what we have been doing, I want to give a brief introduction, even though it doesn't require any introduction. As you probably know, like what kind of symptoms are seen in these patients. So majority of the time, like 88% or more of the times people experience, or the patients experience fever, muscle ache, or dry cough, shortness of breath. So in recent times, shortness of breath is the main problem. I think you must have seen a lot of patients, they are dying just because they can't get the oxygen level enough in the body, and that's what the main problem is right now. So along with that, there are some people who don't get any symptoms, like they're asymptomatic. And in my opinion, or many other scientists who believe that why there is so much spread, those asymptomatic people are driving the infection, because people don't know who got the infection and they keep meeting other people, and that's how this infection is spreading so fast. And that's what happened, at least in the beginning. Right now, the proportion is so high. The number of people who have the virus is so high. I think zero prevalence is over 50%, and it will keep going up. And hopefully, over time, I think we'll reach a plateau, and it will start to come down from there on, and we'll have herd immunity. So in terms of the virus, as we all know, the name comes from a coronavirus, and it's because of the crown-like spike glycoprotein, which is here. And that's where the name comes from. Initially, it was labeled or named as novel coronavirus because there was SARS-CoV-1 in 2003, and that's when it was restricted to a certain part for China. US, we did not have too many cases. But SARS-CoV-2, of course, it has similarity. That's why it's named in the same family. And this is the electron micrograph of the actual virus, how this looks like. This is a viral particle and a spike on the surface. So as you all know, that SARS-CoV-2, I think is still a big problem, especially in India and Brazil. These are the two countries right now. The numbers are expanding. And this was a news clipping from New York Times, which was today. And it seems like the majority of the cases in India is because of the variant which is circulating in India called double mutant, and now they have triple mutant as well. Probably, if you know or follow the surveillance of SARS-CoV-2. So the variant, I want to give a brief idea why there is so much variation or so much variant evolution. So I want to give a brief introduction of how the virus goes in the cells and what happens and why it accumulates to its mutation. So if you look at here on top, this is the viral particle and this is the cell membrane. When it attaches to the receptor, ACE2 in this case, it fuses and comes to the inside the cytoplasm, and that's where it uncoats. And the mRNA or the RNA, which is a positive sense RNA, it makes the protein, and these proteins code for polymerases, proteases, and they lead to replication of the virus, and many copies of the viral copies are made, or viral genomes are made. And this process is driven by RNA-dependent RNA polymerase. That's the polymerase which helps in copying the mRNA or the RNA copy of the genome. And since this is a polymerase, it's an enzyme, this has been used or has been targeted by different antiviral, like remdesivir here in one of the antiviral, which is currently in use. But beside that, many other antivirals which target this RDRP or RNA-dependent RNA polymerase, and it blocks the replication. But in this process, when the virus comes in, it replicates, it generates copies, and this polymerase complex doesn't have a good proofreading activity. So we know that if there is no good proofreading, virus can accumulate those mutations, and that'll lead to change in the amino acid sequence. And if the variant comes out with those changes, they are variant, meaning they are slightly different. They're not totally different, but slightly different. That's why they're called variant. And so far, what we are monitoring, this is again a snapshot taken like last week or so, where these variations are happening. So if you look at the y-axis, this is the diversity or the variation in amino acid sequence of the virus. So virus is around 29.5 kilobases long, and this ORF1A is there, ORF1B. I don't know how much you know about the virus. This is like polypeptide, which is coded. Spike glycoprotein, that's where the mutation is causing these variants, and all these different non-structural protein. These are non-structural protein, these are structural protein, this cause like nucleocapsid and all those. So in this case, what happens when virus attaches to the host cell, like ACE2 is the receptor, and virus, the spike glycoprotein, which is primarily like RBD, which is a receptor binding domain, binds here and goes in there. But the mutation, if you look at here in this case, this spike area has the most amount of mutation. If you look at these bars or these lines, they're taller. That means majority of the mutation is going on in the spike glycoprotein. And some of these mutations are highlighted here in green, red, and they are important mutations which have enhanced the affinity of the spike RBD to bind to the H2 receptor. And that's what's causing this, these variants. So why does this mutation occurs, right? So we know that if virus replicates more, virus will accumulate those mutations, and virus coming out will have different amino acid sequence, and some mutation in the spike glycoprotein will change the structure and cause higher affinity. So if you look at this image, which was taken again last week or so, currently we have 141 million cases worldwide. This is again from Johns Hopkins Public Health website. And if you look on this side, that's what is the tracking of the cases from last March till April of this year. So in the beginning, it was like under control, and during like July to October, we thought that we have under control. A virus was replicating with the same kind of numbers were almost similar, like plateaued. But in October till December, the number kept going up. And this was driven by the UK variant. Probably if you know that UK variant is highly transmissible. And that's what drove this peak here. But again, when vaccination and everything came up in January, these numbers start to go down. But again, this number is going up just because these two countries like Brazil and India are having more cases here, and that's what is going on quite a bit here. So if there is a variant of concern or a mutation or a different variant, number goes up. But if you apply the measure, it comes down, and at that point need to come down further to make sure there is no additional variant which is driving these cases. So in the US, if you look at the numbers, there were a couple of peaks, and this was the bad ones where we had over 150,000 cases a day, or maybe up to 250 some days, which is quite a bit. And that's the current number in India, at least official number, like they're reporting 350,000 number of cases each day. But the number are quite down here. If you look at that, currently the numbers came down to the level which was in October or so. But again, there are some increase, and this is primarily because of the UK variant. So where is this peak coming from? The peak is coming from the numbers in India. So this is the amount of reported cases in India. So it peaked around August till September, and then came down quite a bit down. But in recent past, like in the last 2 months, it has picked up quite a bit because I think the social distancing and masks were not properly used, and it lead to more viral transmission, and transmission leads to more variant evolution, and that's what's going on here. And other countries, like I mentioned earlier, Brazil is another driver of these cases. So the numbers were like, it never came down actually in Brazil, and that's where they found this variant of concern called P1. I don't know whether you know about these different variants or not, but this variant can cause reinfection in people who are already vaccinated or have prior infection. And in the UK, the numbers are quite controlled, just because the vaccination effort is quite good, and they had a lockdown, a quite efficient lockdown. In South Africa, there were 2 peaks and number came down. So the point here is that if there is a higher spread of the virus, there will be evolution of viral variant. And it's like one of those things, like when you have higher different variant, it will drive the infection rate. So the good thing will be to control the virus by having physical measures like social distancing and mask and all those. That's why it will be controlled. And here in this more close-up picture of how these changes are affecting the binding of RBD to the ACE receptor. So this top is the RBD of our spike glycoprotein, and this area is a receptor binding domain. It binds to the ACE2, which is in green here. And in more close-up here, some of these changes which are highlighted here by yellow or blue, these changes are causing this protein to attach more efficiently to the ACE receptor. So this receptor change has affected the transmission, and we know probably if you follow the news again, B117, that is the UK variant, it has from January, it was quite small proportion, like 5% or so. It has taken over, like over 50% of the cases are in the US. This is in the US. I mean, the cases are, I'm talking about, these are in the US. 50% of the cases in the US are from B117, and this is the UK variant. And the other variant which we are seeing here is a 429 and 427, and it's not shown here. It should be this one here. These are the variants from California. And these variants, although they are variant of concern, but they are not increasing as drastically as this UK variant. Another variant, if you probably followed UK, or not UK, and NY, New York had reported this variant 526. It has increased a little bit, but not as concerning as B117. So why this genomic surveillance is required, or how did we find out these variants are evolving? So we did sequencing. Our UK is doing sequencing quite a bit. I think they did, at least they did in the previous past. 10% of total cases, what they are getting each day, they sequence them to find out what is there in the community. And that led us to identify these variant of interest or variant of concern. And probably you know what this means. Variant of interest meaning that those have started to increase in number, but they are not yet concerning just because they are not causing more hospitalization or more deaths. But variant of concern are certainly causing concern in both ways, like they are enhancing their transmission or more hospitalization and as well as more deaths. So those are more concerning. And surveillance or genomic sequencing can also identify whether these variants are going to affect the vaccination approach, because if these changes are causing the immunity to be different, they might still evade the immune shield either by vaccination or by prior infection, so the virus can still get in and cause problems. So knowing this variant is certainly important to predict what may happen over time, and also determine the therapeutics efficacy. Like this convalescent serum as well as antibodies are given to patients to treat, and if there is a difference in the spike glycoprotein, those therapeutics might not work very well. And in this picture, I want to show this Indian variant, which was B1.617, and recently from West Bengal, they identified 1.618, and that is triple variant. And these all are categorized in variant of concern, or VOCs. So what we did here in Nevada in our lab, so we do test, like we work with public health lab here. We get all these nasopharyngeal swabs, which are used for detecting the viral load or positivity rate. And that is done by extraction of RNA and sequencing and identifying by phylogenetic tree and what have you, all different programs. And our assay, when we did our study from March 6th, which was pretty early in the pandemic, till June 5th or so. And we monitored the patients in our community or in our School of Medicine, by taking the SARS-CoV-2 specimen from fatal cases as well as symptomatic cases. And in the beginning, the idea was to see what is causing or what is driving the death in these patients. And we wanted to find out the molecular differences between the virus. So we had like a lot of news release, and this was one of the one from Nevada today. He's the director of Genomic Center, and we provide the samples to him. He provides the sequence to me, and we analyze to find out all these variants. So our first study, which was published last year, was to determine whether there is any correlation of variant or SARS-CoV-2 from fatal cases or people who died. So we found that there was no good correlation, not any specific cluster was causing death of these patients. So by the way, the death or fatal cases are in red here, and symptomatic cases are in black here. So there was no clear indication that any specific variant is associated with death. But again, the case at that time was different, and now it's different. And we are trying to analyze these cases here in Nevada to find out whether B117 is causing are more fatal or causing more hospitalization or not. So those are ongoing studies. And the other study, like Somesh mentioned earlier, this was the major study which we found out because we were screening these patients during early in the pandemic to find out what kind of variant they got. And we found one patient who was reinfected. Again, this was in last year, like from March till June. And at that time, reinfection wasn't known because we thought that if somebody is getting infected, they should be protected because their B cells and T cells, they both will be, they will mount immune response, and the person will be protected. So this was highly significant to show that, okay, no, it's not the case. A person who recovered from SARS-CoV-2 infection can still get infection. And this was the first study in North America. And there, several study was published from Hong Kong, but they were not very, they did not have a clear indication of molecular level. And that's why we managed to publish this in Lancet Infectious Disease. And this was covered like on all major outlets like CNN Health, Science Magazine, Washington Post, and Forbes Magazine, even Time covered as well. And so what we found in this patient was that this one person who is 25-year-old male, when he came to clinic, he did not have any underlying condition, meaning his immunity was good, his B cell, T cell, the numbers were good at least. He got sore throat, cough, headache, nausea, and diarrhea. These are original symptoms or classic symptoms of SARS-CoV-2 or COVID-19. But after some time, he recovered. Like in 10 days, he recovered. And here, if somebody recovers, if they have to go to work or so, they need to be tested negative. So he was tested negative 2 times on May 9th and May 26th, and then he was allowed to go to work. But after some time, like from May 26th to June, or May 28th itself, he started to feel similar symptoms. In fact, his symptoms were like even more severe. Like he had dizziness, and he was hypoxic, meaning he wasn't able to breathe. So he needed hospitalization. He was admitted to ICU, and at that point, his saliva was taken to test the SARS-CoV-2 and came out positive. And here is a CT value of the viral load. And he was also tested with IgM and IgG to ensure that his immunity was mounting, and he was getting enough immune response. And so he was like immune competent. He was not like an immune compromised individual by some other infection or so. So when we sequenced this virus was quite different between these two events. Like first event was here and reinfection was here. So they were far apart in phylogenetic tree. And what does it tell you? That the virus was not the same. Like he actually got the reinfection. He wasn't like maintaining the virus all along. If he was maintaining the virus, it will be similar. They will be not this far apart. So this confirmed, or this provided the molecular definition or molecular characterization that this person was in fact infected with 2 different variants. And at that time, there was no classification of variant. We were, I think, smart enough. I don't know. We were good enough to call these different kind of virus which was causing these reinfection. And when we tracked a clock tree, so here case A was the first infection, case B was a second infection or reinfection. If you look at where they diverged fairly early, meaning if the virus evolved from this virus, this branch will start from here, but no, the branch started fairly early, meaning that the virus was in 2 cases that were diverged, or they came from different origins, and the divergence happened fairly early. And like I said, we are still analyzing a lot of reinfection cases here in the US and in India. I know I did talk to a friend of mine at JNU. He studies COVID, and he did say that they're getting a lot of healthcare professionals like nurses and doctors, even though they have vaccination, but they are getting COVID second time. So I told him, send me the sample out of sequence to find out what variant is there, because we are already doing those kind of things here. Although the number of cases are very low here, but whoever we are finding, if they're getting infection after other vaccination or when they have prior infection, we are characterizing the different variants. And here is the one case I'm presenting just to show that what is trend now. And these red lines at the bottom, by the way, this is sequencing of entire genome, a good coverage. And these red lines are the variation, which is different in these 2 cases. And these which are similar are marked by asterisks. So if you see majority of these mutations or these red lines are different between these 2 viruses from first infection and reinfection, it tells us this virus is very different. And we did look at the lineage, and they're in certainly different clades or different lineages. Another story what we had from those sequences where we found that the specimen in Nevada, what we picked up, by the way, this is probably you all know, US map, and these are different states, and this is Nevada right here, right next to California. So we found this unique variant which had this amino acid 323 mutated to phenylalanine. And this was nowhere, not even any other state of the US or even the world. We looked at every sequence submitted to GSAID and we found no sequences with this phenylalanine at 323. So this is pretty interesting. And we published this in Journal of Genetics and Genomics this year, like last year or so, this unique mutation in this one. And this unique mutation is important because if you follow the spike glycoprotein, this was the first mutation in spike glycoprotein, D614G, they call it DUG. And if you look at the pattern when it changed, so this is like January of 2020, and D is this color, and teal color, and G is the yellow color. So the transition happened quite early, like February and March, it took over, and currently majority of the SARS-CoV-2 circulating in any country is with G at 614 amino acid. But what we noticed that this amino acid, which we picked up here, phenylalanine or 323, it changed from proline to leucine, almost same pattern. This is two different ways to show it. This is like a clock where you show like how these different viruses are evolving, and this is the overall change. So if you look at the pattern, it's totally identical. So this was pretty interesting, but in our case here, we saw this was phenylalanine. And D614G, if you remember, or if you have seen the paper, when they changed this D614G in a lab setting, there were more virus production. Like if just single amino acid change at 614, it caused more variant to be made, and that's what drove the higher infectivity. And same thing is going on with this B117 variant right now. And there are no clear evidence though, but the patient data does show that, yeah, there is something going on with this mutation. So where is this mutation 323? This is in the interface domain of RDRP. So if you remember the genome, and this is the crystal structure of RDRP along with other accessory protein. And we assayed the RDRP activity, and we saw the leucine has slightly higher level of activity of RDRP, and phenylalanine is certainly slightly higher than wild type, which was in Wuhan strain. And this was also changed to histidine, which is no change. So the point here is that these single amino acid changes are causing these proteins to behave differently, which is expected, I guess. So where are we with this mutation now? So if you look at the global picture of this variant 323 from leucine to phenylalanine, this is enhancing or increasing in Africa somehow. Some of them are in Europe as well, in Asia as well, some of them. But majority of these increase with phenylalanine is going up in African continent. And here is the relative proportion of this one. So PL, you already saw that. And here is the blue, is the phenylalanine. It's kind of a lower proportion, but still going on. And we want to monitor like what this is doing to the virus variation or the variant evolution. So you saw here that over time it changes, the pattern changes. So what we need or what we think is need to be done is like a real-time monitoring of SARS-CoV-2, because if virus replicates in over time it changes to something else, we need to find out right then, not later, when the virus has spread so much. So what we do for real-time monitoring is this individual level testing where people's NP swabs is taken, RNA extracted, qPCR is done, and from there on, sequencing is done. But along with that, we have also used this surveillance system from wastewater, because we know that if a person is infected, this virus is shed in the stool, and that is collected, and we take that wastewater, not we, but like our collaborator, they provide RNA from wastewater, and we sequence them to find out what is the snapshot or what is going on in the community by sequencing, or what signature we see in the community. And this individual level testing let us identify the first UK variant in the beginning of January, later January, like January 25th. And all these news report, because we work with public health lab, they quickly report to our news articles, the newspapers, and they publish it when we detect any of these unusual variants in the community. So in our effort of real-time monitoring is from January till April 5th, this data till April 5th, and we have additional data. I did not update this one. So if I look at this variant, so again, these variants are, like right now, they're like thousands of variants circulating. And these variants are a minor change in the spike glycoprotein. That's what's causing these variants to happen. So we have more than 40 variants in Washoe County, our community. And we monitor every day, or at least get some samples every day, and P2 and all these variant of concern we monitor. B117 is monitored. So right now we are seeing a decreased level of virus in the community, which is good. And this was, by the way, put together by a master's in public health, Dietrich. She uses this data and provide this information to us for analysis and so on and so forth. So UK variant. So I will not talk about other variant because there are so many variants, but the variant which is concerning, like B117, which is the UK variant, and this is, by the way, Nevada state, and these are the counties, like this is our county, Washoe County, this is Clark County, that's where Las Vegas is, and Carson County here. So red is the B117. So if you look at these pie charts, some of the counties like County, Carson, and Washoe, the one we are in, had quite a bit of B117, which is the UK variant. And when we monitor the spread from whenever we detected in September 2020, we had one case. In November, we got one case. And in January, by the way, these different color coding indicates that with increasing week, like week 1, week 2, week 3, this circle goes bigger. And January is in green, February is in blue, and March is in red, and April is in magenta. So if you focus on this, just look at the color. We don't have too many blacks, so they were just beginning to have these UK variant. November, we had just one case here. In January, we had a couple cases, so there was not much increase within those months. And February, we started to see a couple more, like here. But in March, it's totally exploded. So if you look at the majority of these dots or circles are red, meaning these cases appeared in the month of March. Even though that this data is for just 2 weeks, you see there are so many dots. And by the way, each dot represent as individual patients here in Nevada. These are de-identified. I can't provide the more information, just a dot. That's how we represent them here. So the point here again is this UK variant started to explode from March till April. And this is going on. We are continuously monitoring, and we keep updating these charts. And this is a pretty interesting slide. Like here, so I don't know whether you know what California variant is called Cal20C, which has mutation at 450R, and we have quite a bit of Cal20C. Just because we are right next to California, we have see the pies here. This county is like more than 50%, and Clark County, where the Las Vegas is still quite a bit there. But importantly, we have Brazilian variant, but not too many though. Brazilian variant is right here. These two color are the Brazilian variant. And with quarantining, we managed to contain this. In entire state of Nevada, we did not have too many Brazilian variant because we identify and quarantine them. They're not allowed to come out. They just we can suppress the spread of those variants because this is highly infectious and it can evade the immunity. So this is bad one. South African variant, again, same thing. We got just one reference case, and this was a travel-related. Our healthcare professional, they found out this person had traveled to South Africa. He came here, and he brought the strain. So he was quarantined, and there was no more cases. But recently, what we found is this double mutant called B617, which is currently highly prevalent in India, like these cases. So we have these one here. Just because I think somebody traveled from India or something. I don't know what exact story is, but they detected this Indian variant in California, like San Francisco. We are so close to San Francisco, and we think that these cases are travel related or so. And this double mutant, like I say, this one is a mutant, a mixture of Cal20, which is California variant, plus Brazil and South African variant. Again, probably you don't remember these numbers, but P1 is the Brazil, 135 is from South Africa. But the good news is that the Covaxin, I don't know, we must have followed this news that Covaxin came out and they said that, okay, there's a data shows high protection against all these different variants. So these Covaxin and Covishield both seem to be protecting these different variants. But again, we don't know the efficacy, the exact data. These are like patient data, and there's no in vivo or neutralization assay done. So that's one of the things we are doing. I am interested in figuring out whether the patient serum is neutralizing the variants, California variant, even Indian variant I got in our lab, and we will be testing the neutralization to see what kind of efficacy they might have. So another part of our surveillance is, like I said, wastewater. And the wastewater, like here, everything Seaver line is connected. You can connect or collect from the interceptor the wastewater, and you can analyze the genome copies. And this study was done from July 1 till December of last year, or December 2022. And here, if you look at the daily cases, so this gray line is daily cases in the community. And N1 and N2 these are the genes for SARS-CoV-2. So they did correlate pretty well. Like if there is a high number of daily cases in the community, there is a high spike in the wastewater. So they correlate. Bottom line is they correlate very well. The amount of virus present in wastewater correlates very well with the cases in the community. Since we work like with multiple agencies, we can get all these data and make sense of the data. And recently we did a sequencing of wastewater sample from January to April. And what we found is that spike glycoprotein. So this is, again, I highlighted or demarked the area of spike area, which is this area. And if you look at these red lines, this shows different variations. And these variations are the defining mutation for different variants. And based on that, we know that there is a S501Y that is present in UK variant, and B1351 signature like E484 and P.1 variation. So all these signatures are detected in our wastewater. But again, we are not seeing too many cases of these in the community. So maybe there is some level of circulation going on, we don't know. But that's why we got to do both, like active screening as well as passive. Passive meaning wastewater, where nobody knows what's going on, but at least you are getting a snapshot what is happening in the community. So with that, what we matched is that we try to get the signature sequences of wastewater and from the patient. So here, NP swabs, meaning these are patient specimens, like small dots are patient specimen, and larger dots are from wastewater. So this one, this one, this one, this one, and they are put together on this Kimmer tree. Maybe this is too complicated for you, but look at just the color. So the blue color is the UK, California variant. Wherever you see blue outside, this is pangolin lineage, that's the California variant. And this is the collection date. The orange color is collection date. So I took just for January first week. And if you look at the amount or the color of this, the California variant, here you see blue, and this is present in the wastewater. And not all the facilities showed that. We compared 4 or 5 facilities, like this one here are different dates as well. So some of them did not have the signature, but this one, TM, like we call Truckee Meadows, this had the signature of California variant. So the point here is that the wastewater is a good source of getting a snapshot what is happening in the community. And the changes what we saw was like S13I that is there in California variant, W152C, this again, the spike signature, 450R, spike signature. Besides that, there are other mutations in ORF1 and ORF-B as well. So this shows a good correlation of screening in patients as well as wastewater. So where are we going with all these? Like these variants, like we are detecting these variants, but whether they are causing severe symptoms or not. So earlier we had this paper published in Gut where we showed that people who have severe symptoms of SARS-CoV-2, they had diarrhea. And what was causing diarrhea? We found out the serotonin level, which is an enzyme. It is present in the plasma of those severely or critically ill patients. So don't look at the data. I just wanted to show that, okay, we looked at different symptoms like nausea, abdominal pain, vomiting, and diarrhea. So out of these 10 patients, they had a good or high level of serotonin in them. And serotonin is depicted by HT. And also one thing we found out that IL-6 was quite high, which we know, right? Cytokine storm is caused in those patients who are severely ill. And the patient without diarrhea had lower IL-6, and healthy control had no. So there is a good correlation that some of these variants are causing or driving these pathways, cytokine storm or diarrhea. And we want to further take more patients in our study and see whether there is a specific variant associated with this phenotype or these clinical signatures. With that, I would like to thank all the members who are working with us on this study. Mark Penduri, he's the director of Nevada State Public Health Lab, and Michael Picker, he's from Southern Nevada, Las Vegas area. They both are the director of Public Health Lab. Cyprian Roggetto, Dave Aquine, they're from microbiology. He's chair of microbiology at UNRMed. Paul Hartley, he's the director of Genomic Center. Krishna is a chair of civil and environmental engineering. Sunil, cell physiology, and all these people, and certainly for funding Vice President of Research and Innovation from UNR, microbiology, and their team. And here are some of these news clips where CNN Health, Time Magazine, and Forbes, they reported the reinfection cases. And I will stop here. In the beginning, by the way, I forgot about the graduate course where Somesh mentioned about that. I'm director of the cell and molecular biology program. So yes, I encourage the students from different universities in India or other countries to apply in our program. If they're competitive, we'll certainly like to get them here to be part of our PhD program. Okay, I will stop here and I will take questions. I hope it was not too long.
Thank you. This is an incredible lecture, and there is so much information available out there that we need people and experts to make sense out of it. And thank you for your lecture. I think there are, among your audience, there are apart from faculty members, there are a lot of students, and some questions have poured in. I will start by asking my own questions. We are focusing a lot on the mutants of the spike protein. I think all of the mutants that you are mentioning are the mutants of the spike protein. What about the mutations in other proteins? And right now the virus is with these mutations, the virus is getting more and more infectious. But what about the immune escape variants? Are we detecting them? Are we looking at them as well?
So yeah, that's a good question, and that's one of the interests we are pursuing right now. So we got all these different variants in our lab, and what we are trying to do here now is figuring out whether they grow faster, first of all, and if they grow faster, whether these other mutations, like you probably know all these already, nucleocapsid has got ton of mutations. And what a code has got a ton of mutations. Only protein which is not mutating is an envelope matrix. That's not too much. And RDRP is mutating somewhat, but not a lot. But yes, there is a question of these escape or making these viruses more aggressive in terms of application. We still need to figure out. And we have systems set up. We have this lab, and we have all these variants. Even like I said earlier, I got this Indian variant because we got in our patient samples and we collected those NP swabs, and we can culture the virus from NP swabs. So I'm getting set up to do all those to see. Like the first question, what I want to address is like why this Indian variant is moving so fast, or why this is circulating so fast. whether the immunity is like playing a role or just the virus is more infectious. So certainly over time, know all those answers, and it's a good question to find out, and we are interested in all those. I'm sure a lot of other people are interested too, but we will do whatever we can in our capacity to get that to you.
Okay, thank you. A few questions from the students. One of them asked that how can we prevent ourselves from new mutants of coronavirus even after being vaccinated? Can vaccinations ensure protection from new mutants by coronaviruses?
So far, studies are suggesting that it's not panic situation. Don't push the panic button just because, there may be slightly lower protection, but at least there will be lesser or no severe symptoms. You might still get infected, but the severity of the symptom will be very, very low. That's what we need to do right now. We don't need this much fatal cases or death. That needs to stop. And that will stop by vaccination. But over time, the vaccine will change. I think we'll have to come up with a different combination of vaccine. Like we have to incorporate these different mutations to enhance the efficacy. But right now, the need of the hour is to make sure that we are getting everybody protected to some level. Maybe not 100%, maybe 80%, 60%. And that will cut down viral spread because we don't need this to go wild and mutate more and create more mutations. So suppressing or reducing or plateauing the curve is very critical right now, and that's what will help us. And the vaccination is going to help. It will provide enough protection to be not severely sick. That we know. But beyond that, I think whether you will get virus or not, we don't know that part because these vaccines are efficacious, but maybe the efficacy is lower. We don't know. Those studies need to be done, and over time, a month or so, we'll find out. So that's my recommendation: get vaccinated and use mask. I think that's a good protection, right? Everybody, Fauci here keeps saying that, double mask and all those. If you go in public setting where you feel like there's so many people out there, even if they're not symptomatic, you got to have these masks on. Make sure that you are not inhaling the virus. And major route of infection, as we all know that, is through nasal. Our mouth can take it too, but nasal, like respiration, a respiratory droplet is the major source of infection. So try to prevent that, and that will certainly reduce down the chance to get the virus.
Regarding, I think at one of your slides you showed, you mentioned something regarding the collection of plasma from infected patients, and then looking at that, is that an option? I think a question has come up by one of the students. Has there any significant therapeutic effect of convalescent plasma therapy been observed on coronavirus mutants? Because there is the systematic reviews have often shows that convalescent plasma therapy is not effective. But what is your experience? What do you suggest on this?
So yes, exactly. That's what we are setting up to do. So what we are collecting, we are collecting serum and plasma from people who are vaccinated and people who had prior infection. And we want to compare with these different variant neutralization at least to see whether their antibodies are neutralizing enough to prevent infection. So that data, I don't know, but it may provide some protection, but not. High titer IgG will be better than the convalescent serum. That's what the studies has came out. Like India did a large scale study, right? It was published in Science or something, like pretty high impact journal. Like they had multiple centers and they analyzed the effect of convalescent serum, and there wasn't any positive or good effect to prevent infection. But again, this purified antibodies or high titer antibodies are certainly helping. And these companies maybe don't Regeneron and all, they are making these high titer antibody therapeutics, and they certainly are helpful in critical cases where Donald Trump was injected with all these things. I don't know whether you follow the news or not. When he was sick, he was injected with all these different therapeutic antibodies, right? He was given everything. They are protected. But again, over time, they might not be because the epitope changes. Those antibodies are monoclonal, kind of monoclonal. They will not recognize, and the efficacy will go down. So the mutation is a problematic thing right now, but if we have a combination of a cocktail of these antibodies, they will certainly be useful in preventing.
Does Professor Ramarao have any question? I think he wanted to ask a question. If Professor Ramarao, you have any question, you can unmute and ask.
Professor Shiva also.
And Dr. Shiva also. Dr. Shiva Krishnamurthy, do you have any question?
No, thank you very much for your nice actually presentation, sir, Dr. Varma.
Thank you.
Very good. It's very good actually. I have to rearrange my slides automatically. Okay, thank you.
Okay. I'm sorry if you had to do some arrangement, adjustment. I'm sorry.
No problem. No problem.
So here I would like to add that both our speakers are from BHU. Both are PhD from BHU, Professor Ramarao as well as Dr. Subhash. So we have BHU alumni today with us.
Yeah, I'm happy to hear that.
Yes. Sure. I worked in VHU also for a long time.
What year were you there? Like
I'm a student of VHU from 1973
Wow.
to 79 in IT, pharmacy. Then subsequently I joined the department and I worked up to 1990.
Yeah, no, I went there 96, 97 or something like that, and then came out. And
Yeah, good. I'm more or less a senior citizen now. I retired everywhere.
No, that's okay. All good, right? I mean, it's just, yeah, it's good to know people from old institutions. I'm happy, I'm flattered to hear that, actually.
Thank you very much. Thank you very much. Nice talk.
Thank you.
Dr. Shiva, if you have your question, you can ask.
Yes, sir. Hope I'm audible. So it's a nice presentation. I just have a question. Like, what is the limitation with the variants? Like, for example, commercially, lot of testing kits validated and approved. So whether really they are able to pick all the variants, or is there any problem with the mutations?
Yeah. So that's certainly a good question. Like you probably know, in India, like I find out, I have a lot of friends there, right? They tell me somebody got tested negative and they have symptoms of COVID. So there is a problem, especially South African, they get the recently the area where it pick up. So it doesn't pick up that anyone, a CDC approved primer, they don't pick up those variants. So it is causing a problem, and over time, I think it will be more problems. So some cases where they will see that some people are showing negative, but they might still have the virus, it's just not being tested properly. So it is a valid question, and this needs to be addressed going forward.
Okay. Thank you, Dr. Subhash. It was indeed a very nice presentation. Thanks a lot for your time. And I'm sure it's about 10 o'clock there. So I would like to thank you. If you want to, stay with us, it would be great. But as it's 10 o'clock there, so it's up to you. So we will have our next presentation. Dr. Pramod, over to you. And thank you, Dr. Subhash. Thank you very much.
All right. It was nice meeting you guys. I will take a leave, okay?
Thank you. Thank you.
All right, bye. Thank you, guys. Okay.
Bye, sir. Okay. And after a pretty good and incredible talk by Dr. Varma, it's my honor to introduce Dr. Sivanathan Krishnamurthy, Assistant Professor, Department of Microbiology, AIIMS Bathinda. Dr. Krishnamurthy has done his MBBS from Government Vellore Medical College, Vellore, Tamil Nadu in 2011 and completed his MD in Medical Microbiology from PGI Chandigarh in 2016. His specialization are bacteriology, virology, mycology, immunology and parasitology. He has also served as Assistant Professor at Department of Microbiology, Jipmer, Pondicherry, and as Senior Resident at Department of Microbiology, PGI Chandigarh. He has also published several research papers in reputed journals and attended many national and international conferences and workshops. Now, I would like to invite Dr. Krishnamurthy for his talk on current evidence of COVID-related immunological markers in clinical diagnosis and prognosis. Dr. Siva.
Thank you, sir. Thank you so much for your kind words. And I really thankful for the organizing team to give me the good opportunity. So directly I will go to the presentation. Before that, I will just make sure it is open. Is my screen is visible, sir? Is the screen is visible, sir?
There is some, no sir,
Oh, not yet. Not yet. Yes.
not yet.
Yeah, we can see your.
Yes,
Yes.
now it is. It's fine.
Okay. Okay, hope I'm audible and my screen is visible. So today's that topic, which is I would like to present. Actually, it was suggested like immunology or immune mechanisms, but as a clinical microbiologist, we have usually certain questions in our mind when the clinicians inquire certain things like how to predict certain severe disease or how to predict the necessity of oxygen requirement or invasive ventilation. So how to go about these things? If you are able to predict those things early, so it will be well and good for the patients. So keeping those aspects in my mind, the topic suggested was current evidence of COVID-related immunological markers in clinical diagnosis and prognosis. So before going into this, I just want to make certain points like what is the understanding on the markers or diagnosis and prognosis, etc. As a lot of undergraduate students are there, so I just want to make sure like what we are speaking. So markers means biological markers. There are biomarkers. So whenever there is a substance, any physiological substance which is present in our body, when it is quantifiable, when you are measuring those physiological molecules, and if you are able to see is there any change when there is a disease progression, if there is any change in those molecules, then you can consider them as a biological molecules. So the biological markers are, so many markers are there. Either it can be RNA, it can be DNA, it can be antigen, antibody, any molecules. If it is there in your body and if it is altered over the period of disease progression, then you can consider them as a marker and it should be quantified and it should be followed up. Then coming to the another terms, diagnosis and prognosis. Same way, diagnosis is if you are able to demonstrate certain causative agent, it can be infection or it can be a non-infectious etiology. So if you want to detect that particular etiological factor, then it is diagnosis. Prognosis is the same way, like how we have mentioned about the biological markers. Based on certain biological markers, if the natural history of the disease, when the disease is progressing, if it is getting cured or if the condition is getting worse and if need certain intervention, so that based on the intervention the disease can be prevented or it can be treated. So for the monitoring of such a thing, if you are having certain factor, based on the factor you can predict the outcome, so that is called prognosis. So with this brief background, so we will go into the topic. So I think already Dr. Varma described the morphology and the pathogenesis aspect of the virus. I'm not going in detail on those things. But for the diagnostic aspect, what are all the things we are dealing? For example, in genome level, if we are thinking certain genes like ORF gene and N gene, S gene, many other genes are there. So those genes are used as the target for real-time PCR, reverse transcriptase PCR in this viral infection. The same way, if we are seeing certain genomic element which is actually coding for the proteins, especially for the spike proteins. So these spike proteins usually, the spike proteins are main important things which is coding for the different proteins and against which there are antibody development. So the antibody detection is based on this particular antigen moieties over the virus. So pathophysiology, I am not going detail as it is already discussed with sir, but certain points like when the virus is entering into our body. So what is usually happen? It is going into the alveoli. In the alveoli, it is actually starting the like it is recruiting the inflammatory cells. So the inflammatory cells are recruited over the alveolar surface, and the macrophages, alveolar macrophages is the important one, and there will be subsequent release of various cytokines, chemokines, and this particular entity called cytokine storm. So what will happen? This storm will recruit further, like it will increase the vascular permeability of the alveoli, and it will again recruit multiple immune cells. So there will be increased number of immune cells and chemokines, cytokines, and so many factors. So what will happen? So this particular condition usually leads to cell death and damage of the lung parenchyma. Whether this cytokine storm syndrome, usually it is mediated by interleukin 6, interleukin 1 beta, and tumor necrosis factor. Whether these things is only seen in this COVID virus? No. There are n number of viruses actually playing role and making cytokine storm. Like in the past, there are reports of like influenza virus pandemics, or the SARS-CoV-1, or Middle East respiratory syndrome coronavirus. So other beta coronaviruses also causes the cytokine storm syndrome. So what is happening, like whether all the chemokines or cytokines, whether they are same in all these infection, so that is makes sense. Like here, there are different infection will behave differently, and they will make different chemokines, cytokines, and their expression. So today's topic is based on this immunological marker, whether how much they are expressed, how much they are deranged, whether it will be it will help in the decision making in the clinical side. So that is the topic today. So first we should know what are the investigations available in the clinical setting. So there are whenever we are speaking about microbiology, usually we will consider microscopy, culture. But in this condition, any viral infection, the microscopy and culture, it is very difficult. As we know, it need a electron microscopy, and for the culture, you need a tissue culture setup. So it is not possible to make such a setting in all the laboratories. It is feasible only in the research level laboratories. So in a normal diagnostic labs, is it possible? No. So what are the things available currently? So the one is real-time reverse transcriptase PCR. There are two things are there, qualitative and quantitative. Why I want to mention this particular things here? The quantitative is very important. Why? If quantitative is available, then you can make the viral load, and then you can predict the disease severity and the outcome. But though it is a real-time PCR, though we are making decision based on the CT value, it doesn't mean that we are actually giving a quantitative result, because it is a qualitative one. We are giving the result as positive or negative based on the cycle threshold. Why we are not able to predict the viral load in this thing? There are many factors affecting the viral load, like from the point of sample collection. If the enough material is not taken from your nasopharyngeal swab, from that point to transport condition, storage condition, and extraction procedures. So many factors will limit the detection of the viral load. So what we are doing all over the world now, currently recommended method is reverse transcriptase PCR, but which is actually a qualitative rather than quantitative. Then the next test is antigen detection. It is a part of viral protein, so nucleic acid protein. So it can be detected, but if you are using this particular thing, only you can tell the viral particles are there or not. But whether they are viable or not viable, it is difficult to tell. Again, it is difficult to tell the viral load. Then coming to the antibody levels, there are different types of antibodies, and different time period it will be elevated, and it will be remain same for a long period. It's difficult to tell whether it is a recent infection or past infection. Then coming to the radiology, which is the very important thing, because it can predict the disease. So when the CT scan, if you are doing in the patient, so the lung, which is the main target of the virus, so the lung parenchymal involvement, how many lobes are involved, how much of disease is progressed, whether just pneumonia is there or there is a fibrosis, which is irreversible thing. So these kind of things can be made through the radiological investigation. So when we are doing those investigations, you can predict whether the patient can be reversible level or irreversible damage is already occurred. So that is one point in this thing. So in none of the microbiological thing can predict those kind of thing. Only the radiological thing can predict that particular aspects. So already mentioned like PCR limitation, it is not able to detect the viral load and the time at what time you are actually doing the investigation. Suppose a person is exposed to the infection, then if he is tested immediately, will he be negative positive? It's difficult to tell because immediately he cannot be positive. So it needs certain time period. The virus should enter, multiply, and it has to manifest. Then the enough number of viral particles. That is the thing we know as a molecular biologist. People know the lower limit of detection for any instrument. So the currently available techniques also there is a limitation. So usually at what time period the person may come positive? At the time around after 5 to 7 days of exposure and symptom, usually they are coming positive. The probability to get positivity high around the 5th day. If before 5th day if you are giving sample, the probability will go down. The same way after 7th day if you are giving the sample, again the probability will go down. That means that getting the result positive after the when the days are increasing. Even why it is important? Suppose a patient is getting infected, in the 5th day he is coming positive, and if you are actually want to check the severity, if you are doing the PCR in these patients subsequently, is it possible to predict like continuously the viral load will be there? It is not there, but the disease progression may be there because this COVID is a immunological phenomena. Like the pathogenesis mainly because of immunological reaction rather than as such virus particle. Virus also causing damage, but the major damage is caused by your cytokine storm. Then one of the student researcher asked a question in the previous presentation, how long the virus can be there after getting infection, how long the patient can be positive. So there are papers are there, like when you are seeing the positivity, it depends like whether the same person is shedding the virus continuously. That is also possible because the same person, whoever is getting positive, intermittently they can shed the virus. So the PCR positivity can be there even intermittently. The person may be positive, then again negative, then again positive. So you cannot rule out whether it is a new strain getting the infection or the same virus is continuously shedding. So it is very difficult to tell unless otherwise you are doing the genome sequencing. The same way, how long it can be shedding. So the person can be positive from the PCR even up to 80 days or more. So it is very difficult to tell whether if I am getting positive within 80 days, if I am getting positive, it means whether it is a same infection or it is a reinfection or still he is having the viable thing virus or something else. So it is very difficult to tell based on the real-time PCR. The same way antigen same, it is kind of again the viral envelope proteins, right? Nucleocapsid protein. So the protein also can be shed for some period of time. So presence of just antigen, you cannot tell that the viable virus is there and it is actively replicating there. So you can label them as infected, but you cannot tell the disease severity. But if you are doing the antigen test based on a certain platform, like if you are using a quantitative one instead of qualitative one, like whatever commercially available kits are like point of care test, when you are adding the nasopharyngeal swab diluted fluid, then you can detect the antigen, means that monoclonal antibody attached with the antigen, then it will give the positive or negative, that much result. But when you are going for a high throughput instruments like chemiluminescence, if you are able to quantify the antigen, then it is easy to tell whether still the patient is having detectable level of antigen, whether the patient may go into reinfection or whether he will continue, he may progress to recover fast. Everything can be done. Like certain in paper they are showing with the viral load when they are matching. The viral load is coming undetectable, then detectable, undetectable. But when you are seeing the antigen level, there is a linear fashion of decreasing in nature when you are using with the quantitative techniques. Then coming to the antibody levels. So antibody levels, initial 2 weeks, there will not be much sensitivity or specificity. But after 2 weeks, the sensitivity and specificity of the antibody-based tests are high, like IgM or IgA or IgA. Any antibody, it is usually after 2 weeks, it is easy to, like their sensitivity and specificity will be good. Usually we will think IgM usually predict the acute infection. But here in this infection, the IgM also cannot differentiate between your acute infection or past infection. So that you should remember. So if you are able to do neutralizing antibody, then it is well and good. Then it can be useful in the response in the severity of the patients. Like for example, this particular diagram showing the apparent pneumonia in the blue color, subtle pneumonia, that is less severe pneumonia in the green color, and asymptomatic infection in the red color. So if we are seeing in the when a person is having very severe pneumonia, they are eliciting more amount of neutralizing antibody. So in those patients, when you are testing 2 months, there will be higher amount of neutralizing antibody, and when you are testing at 5 months, still relatively the equal number of neutralizing antibodies are there in those patients compared to subtle pneumonia or asymptomatic patients. In these patients, the antibody, that neutralizing antibody levels are not that level to tackle with the infection. So that is the importance in the neutralizing antibody detection test. So what are the antibodies like? Now from here onwards, I just want to show certain systematic review with meta-analysis, because the what are the important markers actually raised or decreased. So many of the students may aware, as working in university, you people aware about the systematic review and meta-analysis, and also the interpretation of the forest plot method. Just those who are not aware, just you just see in these plots, there is a line, central vertical line, that is a mean, and whatever values shown in either of the side, it is a deviation from the mean. So this much you should know for this time sake. So if you are seeing whatever towards shifted towards right side, it means that increased in level. Whatever left side, that is decreased in level. So I am speaking only about the severe infection hereafter. What are all the markers which is elevated or decreased in the severe infection, I am going to speak. So in this aspect, like specific antibodies, IgA, IgG, IgM and IgE, the IgA and G, it is elevated in the severe infection rather than non-severe infection. At the same time, IgE, there is no change between the severe and non-severe infection, but IgM is reduced in the severe infection. So this is one good systematic review, because there are so many systematic review articles are there, but either the number of studies included are very less, or the number of markers included are very less. So this is a good systematic review which shown around 32 circulatory immunological signatures they have included. All the things like cells, cytokines, antibodies, and other cells, like apart from T cells and B cells, other cells in the peripheral blood, everything it is included, and what is happening in there in the patient's blood, whether severe cases what is happening and non-severe cases what is happening. So based on this systematic review and meta-analysis, we are seeing the interleukin 2, interleukin 2 receptor, interleukin 4, interleukin 8, interleukin 10. All these markers are elevated in the severe cases. Then TNF alpha also increased in the severe cases. But interleukin 5, interleukin 1 beta, interferon gamma, there is no change in severe or non-severe cases. But the recent systematic review, after this particular systematic review, it is showing interleukin 1 beta also increased in certain critical cases. I will show in the later slides. So why I am keeping the interleukin 6 separately? Interleukin 6, which is very, very important marker, which is very much responsible for the cytokine storm, and there are n number of studies favoring for the elevated levels of interleukin in severe cases than non-severe cases. So this is a potential marker which can be utilized in the prognostic aspect rather than diagnostic to predict the severity and to predict mortality or survival rate. The interleukin 6 can be a potential marker. So till now, what all the evidences are there with the marker? Like we know it is an inflammatory marker, and also it is stimulating the acute phase reactants such as C-reactive protein, fibrinogen, etc. Even this C-reactive protein, LDH, etc., they are actually currently they are using in clinical setting to predict the severity. But interleukin 6, it is not used that much level, but research aspects and certain institute they are doing this particular markers. So will it be helpful like this, if you are able to detect this marker early, so it can predict the risk of mechanical ventilation, or if you are able to detect the marker, it is able to whether we can use this particular level to guide a monoclonal antibody therapy or some other therapy. So there are certain evidences are there. Like for example, we usually like monoclonal antibody, that tocilizumab initially in the pandemic when it is started, there are a lot of papers showing tocilizumab is very effective. Then over time there are contradictory papers are started to come. So what could be the problem? Like if we are seeing this this paper showing instead of timing of the treatment, the level of interleukin 6 is very important. As we know, this one is actually inhibiting the interleukin 6 receptor and it is inhibiting the cytokine storm. So if there is already the storm is uncontrollable, then there is no point in starting tocilizumab. That is the important aspect in this thing. So if a patient is presenting to a clinical setting, even he is like clinically he is not looks very severe, but if the interleukin 6 levels are very high, then if you are starting the tocilizumab, it will not able to control the storm. So that is the basic principle behind this particular paper. So what they found, if the inflammation level, that interleukin 6 level is less than 135 picogram per ml, then it is, if you are starting the tocilizumab in this condition, then it is having a favorable outcome than more than 135 picogram per ml. So that is the gist. Like instead of timing of treatment, the level of inflammatory marker is very important to guide the therapy. And the same way, the different levels of this particular cytokine, that interleukin 6, it can predict at risk of severe cases or at risk of mortality. And also it is like when a patient is presenting, nowadays the clinicians, what they are using, like the important measure which is able to predict the severity, it is SpO2 level. The pulse oximetry they are using, they are checking the SpO2. But the thing is, may not, all the patient may not having the decreased oxygen saturation, but they are having the impending problem, like maybe they will develop into decrease in oxygen saturation. So if your patient is coming to the clinic, if they are checking the SpO2 is now normal, but if you are checking the interleukin level, if it is more than 24 picogram per ml, then you can think this patient may develop into, or they may go into the oxygen decreased, deranged oxygen saturation, and they may need mechanical ventilator. So these kind of, these all are very simple single studies are there, but there is no a clear cut like validated studies, or there is no such criteria are there. So it need to be developed based on these findings. Again, including this particular marker into the scoring system, like there are certain scoring system called, that is like cytokine, age, lymphocyte level, LDH. So when they are cumulatively making a score of 4 to 13 and they are predicting what can be the favorable outcome, there is certain sensitivity and specificity. So when they are including the interleukin 6 along with the score they are increasing to 5 to 17, like they are giving score 1 to 4 for interleukin 6. 1 point for less than 25 picogram, 4 points for more than 25 picogram. So when they are adding to the score, the prediction of survival, the prediction of good outcome is increasing. The sensitivity and specificity is increasing in these cases. The same way the mortality risk model, they are proposing different models and they are finding a particular model is looking good, and the area under curve, that is, it is very good predictor. Like when the score is more than 0.07, then the mortality will be more over time. When the score is less than 0.07, then the mortality will be less over time. So to decide this score, they are keeping the interleukin level at 86 picogram per ml. So this kind of scoring system should be validated and it should be implemented in clinical practice. Then coming to the cell counts, till now we have seen the interleukin level. When we are coming to the cells, the different types of T cells like CD3+, CD4+, CD8+, and even T regulator cells, they are actually decreased in amount in severe cases. And also when you are comparing, apart from the cell count, if you are checking the proportion of the cells in the pool, if we are seeing the proportion also either decreased in the severe cases or not altered. Like if we are seeing the CD4, CD8, they are actually the proportion of the cells, they are decreased in the severe cases. Same way, the B cells and NK cells, when you are taking the cell count, the count is decreased compared to the normal range. It is decreased in the severe case. But at the same time, when you are taking the proportion among the whole blood, when you are seeing the proportion, the same B cell and NK cells, they are increased. The proportion is increased in the severe case. So this kind of finding also can be helpful in predicting the severity. But what is happening now? These are whatever studies in a year. What is happened? Like before it is not considered much about the therapeutic aspect. So a recent study in this month published online, if we are seeing like whether any plasma therapy or monoclonal antibody therapy will change this ratio, whatever seen in the systematic review, the increased level, decreased level. Suppose if a patient is going on certain treatment, or if the patient, like for example, there are certain study may come in future, like vaccine induced, like when you are the patient who received vaccine, but they are again getting positive. So in those patients, what are all the changes may happen with these interleukins? So those kind of things, it need to be looked into, because in this paper, if we see, there are certain new markers, like when there is this plasma therapy or monoclonal antibody, they usually going and inhibiting the interleukin 6. So what will happen? The cytokine storm is inhibited. Then what is happening? The normal other, again, this CD4 cells or CD8 cell subsets, again they are getting activated and they are proliferating. So these kind of things. So when to consider these cells are very important. So that is that need to be looked into future. Then coming to the other cells like neutrophil. See, apart from interleukin 6, the another important parameter, that is neutrophil count as well as the neutrophil unit, the proportion of neutrophils. Again, there are n number of studies suggesting increased level of neutrophils in the severe cases. And when we are taking these two things important, like lymphopenia, that whatever T cells and B cells level is decreasing in severe cases, or neutrophils levels are increasing in severe cases. When we are considering more than 7 days, these changes are happening, like lymphopenia or the neutrophilia happening more than 7 days. So it can predict the outcome of the patients. Then coming to real comparison, like whatever findings are there, the hematological parameters analyzed and they made it into systematic review. But the thing is, when you are comparing actual pathology, like when you are able to compare with the radiological diagnosis, as in the first slide I mentioned, the radio diagnosis is very important, because you can see what is the real change in there, how many lobes are involved, how much damage occurred in the lungs, and whether it is whether reversible damage or irreversible damage. So if you are considering all these aspects, if you are seeing here, so there are different groups there, like asymptomatic or subclinical infection to severe infection. The radiological changes are there. But when you are seeing the same leukocyte count or the lymphocyte count, there is no difference between these groups. So whether really it is going to helpful in the real clinical scenario, that is again questionable. So it need to be looked into. Then another one component, monocytes, again proportion of monocytes is decreasing in severe cases, but a new paper, recent paper in this month of April, there is a particular thing, monoclonal HLA-DR. So this is basically a immunological status indicator, like this particular MHLA-DR, which can predict the immune level of, like overall immune level of the patient. So if this level is going down, the immunosuppression also will happen, and it can predict the severity of the infection. So these kind of new things also to be looked into in future. So till now what we have seen, different cytokines either increased or decreased or not changed, different antibodies are increased or decreased, etc. But whatever seen in these previous slides, they have made based on two criteria, either severe disease or non-severe disease. It means inside severe disease, there are severe cases, critical cases. It means critical cases means the patients who may need ICU admissions, or they may need invasive ventilatory support, oxygen support, or they are irreversible damage like ARDS, etc. The non-severe cases are other mild cases. So if you want to look into these groups, like among these groups, is there any changes among these inflammatory markers? Then again, it need to be evaluated further. It need a lot of systematic reviews, and for that it need a lot of studies. So one of such study, it is showing like previous systematic review shown that there is no change in interleukin 1 beta. but there it is like severe case, non-severe case. But in this systematic review, if you are seeing, they are actually comparing with mild cases and severe cases, critical case, mild case, critical case, severe case. So when you are that subcomponent analysis, if you are seeing, there is change in like increase in interleukin 1 beta in critical cases. So this is the problem or challenge in predicting the disease using these immunological molecules. So it is kind of a last slide. What I am trying to tell is, like for PCR or for any antibody levels or PCR from different samples like nasopharyngeal swab or stool sample or bronchoalveolar lavage, etc. When you are seeing kinetics, like the viral kinetics based on the nucleic acid material or antigens or antibody level, when you are able to make over time period for the different patients, like symptomatic, asymptomatic, severe cases, critical cases, if we are able to make such a kinetics for individual molecules, then it will be really helpful to predict the disease rather than just showing or making evidences these all are elevated. But how much it is going to helpful for the clinical setting, like based on this level, whether we can predict something, then it will be well and good. So instead of predicting the severe disease, currently I would rather say prevention is better than cure. So the major thing, like few of the patients ask the question how to prevent. The simple techniques, again, wear mask, take proper social distancing, and use hand sanitizer and hand washing. So apart from this thing, it is very difficult to prevent the infection and it is difficult to get rid of the infection. Thank you. I am requesting any questions or any comments or corrections also invited so that I can improve the slides in future.
Hello, sir.
Hello, sir. Can you able to see? Yes.
Yes, sir. So my question is that, like, is there any relation with the viral load and the infection of patient severity?
Yes, sir. That's what I have shown in the presentation. Like what we are doing is not actually a live viral thing, because it can be either already dead virus or it is like immunologically tackled virus. We cannot go with those things. But if you are able to tell the live virus are there, then we can predict the severity. Because now whatever we are doing now in ICMR approved kits, are lot of kits are there. Every kit's using different targets. For example, certain kits using ORF gene, certain kits using N gene, certain kits using different genes. So when there is lot of variation among the kits, lot of variation among the cycle threshold, is it positive, negative, it is very difficult to predict the viral load and the severity based on the available technique, like available kits.
Okay, so thank you.
Dr. Shiva, thank you for wonderful lecture. I think you remain the lone voice of clinicians among a sea of researchers. We are all basic sciences students. So your voice is very important. I have a few questions, and some of our colleague, Dr. Manju Jain, has a few questions. My own question is regarding, you mentioned that the PCR is not quantitative. Isn't real-time PCR considered as a quantitative technique? Wouldn't that make it suitable? The other thing, you have given a range of different inflammatory markers. My question is more like in terms of logistics. This is possible to do these things. It's possible only in tertiary healthcare centers. It's not possible to, primary health centers. So how efficient, how good this would be in tackling the overall disease, given that it's a pandemic, it's not a localized disease, it's a pandemic. So how efficient that would be?
Yeah, actually, that's why I chose the topic, because I am going to speak to the researchers. So I just want to put this word to you, like you have to develop certain things, like in a low resource setting, how can you modify these things? Like, can you make it as a point of care testing? Is it possible to do with these cytokines? That is the main aim of the talk. I want to choose this talk and I want to present to you people. You have to tell how to go about it. That is the clinician point of view, like clinical microbiologist point of view.
One of our senior faculty members, Professor Varma, was asking about, since you're a clinician, what is your opinion about happy hypoxia concept?
See, though I am a clinician, but I am not actually dealing with the patients. Like I am clinical microbiologist. It is very difficult to answer this question from my side, as it is not my domain actually.
Can unmute herself and ask her question.
Thank you very much. So the whole question.
Dr. Manju Jain.
Yeah, thank you, Dr. Nasir. Dr. Krishnamurthy, that was really a wonderful, lucid talk with a panoramic view, focus on the diagnosis part with respect to the different changes. So I would really request you to please, in short, maybe you can just come up with the CT scan based diagnosis. Maybe I missed it, or
Yeah.
because I have something on that.
Yeah, ma'am, actually, basically, in radiological investigation, like it can tell the different stages of illness. Like when we are focusing, the main target of the virus is the lungs, though it is involving any organ, wherever the receptor, AC receptors are there, so it can involve any organ. But the main focus is the pulmonary infection. So in pulmonary infection, so many lobes, so many, like it is a very vital organ. So there are grading in the radiological investigation, right? So there are mild and moderate and severe gradings are there. And also there are damages. For example, if certain level the lungs will involve, it will get infected, and when with the treatment, it automatically recover from the infection. But certain point, it will not be able to reversible, like it will go into fibrosis. So once if it is enter into fibrosis, then after that there is no point in actually any antiviral or any monoclonal antibodies. It's difficult.
Very true.
So for that only actually the steroids are recommended in the treatment protocols. So you, instead of you are like killing the virus, or you should actually stop the storm. Like you have to control the inflammatory changes. So that is the important thing.
So really, like in alignment of what you said, actually I sort of having interaction with some of my colleagues and collaborators. Like in BHU, there's a hospital inside, right? And a lot of unfortunately fatalities are happening there. So I had a word with them, and like in context of where we are sitting, like in Punjab, in Batinda, you are in AIMS Batinda, right? So I am also tested positive. So basically, we are going for the QRT and all that. So people, but talking with that guy in BHU, right, with whom I am collaborating, so they are really not going by QRT. They are going by the scan. Because they see in and around them, including doctors, so they see the extent to which the things have gone into, that lungs, and then they recommend the dosing and the medicines. But that trend I am not seeing in and around, at least in this state. So am I right on that?
No, ma'am. Actually, it is very good to go based on the radiological finding, as it is the only thing now currently available source. It is the only thing can predict this disease severity and outcome. So it is better to go with radiological diagnosis. All other measures, microbiological measures, are supportive evidence. So you can think, because the radiological evidence is kind of, there are a lot of differential diagnosis for the same changes, because lung and radiology will mimic same. Whatever changes happening in the lung, it will be mimic same. But the pathology may be different. It is not necessarily the virus. It can be some other stimuli or some other virus. Anything can be there. So
or the immune associated, collateral damage. I mean, as you mentioned.
Yes, ma'am.
But the thing is, like with these upcoming newer strains and all, people are saying that basically you look, normal, right? In terms of the symptomatic things. And so the scan is the, is the one you should really go for. So in your hospital and in and around at least I fail to observe that, diagnostic thing in place. So what, please guide us actually.
Yes, ma'am. Right now in AIMS Batinda, we are doing both the things, like radiological diagnosis, CT scan is going on, and we are doing the patient, whoever suspected and whoever, when you are like the clinical protocol, like whatever made according to AIMS Delhi and ICMR. So they are categorizing the patients based on the clinical scenario, how they are presenting and their oxygen saturation level. Then apart from that, then you have to go for the real-time PCR, reverse transcriptase PCR. Then CT scan. So all these things, like it is a multi-pronged approach rather than a based on single parameter. So you have to consider all the parameter, then you have to decide how to go about for the patients.
Yeah. Say on a personal note, I'm sorry I'm asking this on this
No issue.
public platform.
Yeah, no.
Like I'm having this throat thing and all from last almost 10 days. It's not going away, right?
Yes, ma'am.
So I'm thinking to have the scan. I don't know. Will I be entertained for that? Because I am basically not having major symptoms, but yeah, it's there.
Yes, ma'am. You can, like nowadays it is even like people are recommending, or even many people are using pulse oximetry in their home itself. So oxygen saturation one is important thing. And the same, like in the previous slide, one of the slides said like it is not necessarily like oxygen saturation now normal, it may not deteriorate, you cannot give any guarantee. So if you want to do, you can do, but it is depends on the clinician. They will analyze you whether you are really need of the radiological investigation or not. So clinical examination is very important element, like when they are auscultating your lung field, like whether you are having the crepitations or other findings in your lung. So that is also one important measures to go before this radiological investigation.
And another thing I got to know is that generally, as per the ICMR protocol and all, it is being suggested to have, the remdesivir and all in severe cases. But things have to be controlled much before that, before it, deteriorates beyond control.
Yes, ma'am.
Again, coming from the scans, right?
Yes, ma'am.
So, anyways, it's a great talk. I really enjoyed and got to know, the clinical correlates. And somehow, I think, in context of the Indian samples, the reports are not still out somehow. So we are really, looking for, because you clinicians are doing a wonderful job on that. We don't have,
Dr. Manju, let us take a few more questions.
Sure. Thank you so much.
One question, Dr. Shiva, is about the utility or the non-utility of tocilizumab, that it is not efficient anymore against cytokine storm. Are there any options available? And the other question, then after you can wind up, about the low levels of platelet that has been observed. Is there any comment? They come from Kavita Singh and Kiran Patil, and they are asking these two questions about tocilizumab and about the low platelet counts.
Yes, sir. Sir, basically, this regarding monoclonal antibodies, we are actually not having any personal experience. Based on the papers, if we have to comment, there are contradictory opinions, but we are actually not having personal experience in our institute right now. And regarding another one, platelet count, there are actually initially I just try to limit myself in the immunological part, but other cells, like whatever, there are again systematic reviews are there for those aspects also. Even platelet related thing, like because it is a thrombotic phenomena, like the source is one is the cytokine related storm, and another aspect it is like involving the vasculature, like it is a thrombotic phenomena. So again the platelet is involving in this. So like when you are seeing a low count, it means that there is indirect evidence, there is inside, there is a thrombotic phenomena is there. So that may be my observation, but maybe I am wrong, but I have to look into what exactly for this answer.
Okay, Dr. Shiva, thank you for wonderful lecture. We ask you to stay back if possible. Yes, Professor Rama Rao will deliver his lecture. And so thank you once again for this great lecture. You are almost our neighbor, so we can come to you anytime
Sure.
we want.
Sure.
So go ahead. I request Professor Dr. Suresh now to introduce Professor Rama Rao.
Okay, so good morning again. So welcome back. Now we are in the third lecture of this morning session of the International Day of Immunology, and it's a great honor to introduce our third speaker, who is Professor P. Rama Rao. And Professor P. Rama Rao, he's from our institution, actually from Central University of Punjab. So he served as a Dean Academic Affair for almost more than 10 years until he got retired recently. So he's from pharmaceutical background, and he received his PhD degree from Banaras Hindu University long time ago, and he also served as a director of NIPER Mohali, and he has received several awards during his research career. So I request Professor P. Rama Rao to deliver his lecture entitled SARS Coronavirus 2 Drug and Vaccine Development. Over to Professor P. Rama Rao. Sir, please.
Okay, thank you very much for the nice talk and nice introduction, Dr. Somesh. So in that particular case, let me actually say a few points before I talk. I am a pharmacologist, not an immunologist, not a virologist, not a microbiologist. With that background, I want to talk about this particular aspect. Okay, why I am interested in this particular aspect is two, because several actually seniors are taking angiotensin receptor blockers or angiotensin converting enzyme inhibitors. There's a big controversy somewhere in the last year, April, stating that these actually ERB, that means angiotensin receptor blockers, as well as actually S1 inhibitors, are actually contraindicated in actually this particular COVID conditions. So with that particular background, what is that particular aspect? From there, I am starting this particular thing. One of the information what I want to give SARS-CoV, because you are actually the immunology day topic is no COVID-19 to combat COVID-19. So the COVID-19 is a disease and it is caused by your SARS-CoV-2. So it is a fast evolving, hence the information and data is changing every day. The number of papers published on this particular aspect, when we talk about it, is somewhere around 1.25 lakhs in the last 16 months. So that comes out to be somewhere around 8,000 papers per month. So with that particular thing, whatever we talk, it is changing every day. With that actually background, we are actually working and information daily. We have to update ourselves in this particular case. Now, where from actually this SARS is coming into the picture, coronavirus? It actually belongs to beta coronavirus family. It is a 4 families when we talk about genus. In that particular case, alpha coronavirus and beta coronavirus are infecting human beings, whereas gamma and actually the delta coronavirus is mainly actually the other animals is coming into the picture. So it is coming from the bat. That is one of the most important aspect. So the bat is a mammal, the flying mammal. In this particular case, when we talk about this particular case, when we say this is having actually the average life of 16 years, and it's having the robust immune system. That is one of the things. It's an incubator of several actually the viruses. The first actually the coronavirus, which actually came into picture, SARS-CoV, that is 2003, and it affected around somewhere around 8,000 cases, and somewhere around death is 774, which is turned out to be fatality rate 9.6%. The next one, when we talk after 9 years, that is in 2012, the Middle East, in South Africa, and the coronavirus respiratory syndrome, it is spread to 27 countries and having actually infected 2,500. Even the small number, the death rate was somewhere around 850 deaths. The fatality rate is equal to somewhere around 34%. So the present one, where from it is coming, the intermediate host, still there are question marks, how it is coming into the picture. We do not go into that controversy right at this moment. Today morning, when we talk about, it is 15 crores practically infected throughout the world, somewhere around the 31.62 lakhs of deaths occurred. That comes out to be 2.1% fatality rate compared to the previous one, which is 34%. The Indian talk about it today's morning when we talk, that is somewhere around 1.83 crores of cases. And death is somewhere around 2 lakhs. It's crossed already 2 lakhs today morning. And the vaccination, already somewhere around 14.78 crore doses of vaccination are administered in India. This is
few minutes. There's some technical issues. Let me check. Sir, you are disconnected. I don't know, sir, you are not there on the screen. Okay, sir.
Is it connected now?
Yes, sir.
Yes, sir. Yes, sir, you are there. Presentation is still not there. No, sir.
Presentation is not there?
No, sir. No, sir.
1 minute. Okay, is it coming?
Not
No,
yet.
sir. Not yet.
Not coming?
No, sir. So can I try it from my? I can put your slides which you have sent me. It's coming now. Yes.
Is it coming?
No, sir.
What is the problem? How is it happening?
So can I put presentation on our system?
Okay, go ahead, 1 minute, but let me try. Actually, it will be easy for me from here.
Yes, sir. Yes, sir. Yes, sir. You try one. Yes, sir. It's fine, sir.
Is it coming?
Yes, sir. Yes.
Yes, sir.
It's fine, sir.
Okay. So now the point, what it is we want to say about it is, okay, I think I'm sorry for the interruption. I do not know what happened. Okay. Okay. So I think I'm talking about here. I do not know when I missed it, but it is a nuclear protein necessary for RNA synthesis. This is actually the structure. Already people are talking about it. This is what I want to say about the ACE2 expression. Head to toe, all the organs are having ACE2 receptor, angiotensin converting enzyme 2. This is one of the most important aspects. It is starting from the symptoms, anosmia, ageusia, which is actually the loss of the smell and the taste, which actually come into the picture. And it is actually in the children. People talk about it. Inflammation of the toe is also responsible. You can see all the parts. One of the most important latest information is talking about it. It is a long-term effects even after the treatment, after the severe cases recovered. The cognition problems are coming, and so many other actually the aspects are actually coming into the picture. CNS effects, including the brain, which is actually the affecting the part. Now, this is already so many people talked about it. I'm not going to talk about it. Reproductive number. This is 2.2 with a confidence limits of 1.4 to 3.9. I think even after the spike also still within this confidence limits will come, only the mean will actually the changes in that particular case. This doubling time also is changing. This is the old data when I was talking about it, not included the recent information. Already the diagnostics The previous speaker actually explained it. I do not want to get into the case. Most important aspect is to comorbidity. This is one of the most important aspect when we have to talk about it. Whoever is actually the immunocompromised people in diabetes, immunological aspects are compromised, and in the heart problems and in the elderly patients, and who are actually suffering from COPD, the disease, in that particular case, we are having this particular problem. These are the comorbidities when we talk about it. And in diabetes and heart, that is hypertension aspects, angiotensin receptor, there are two actually the aspects. One is ACE2, we are talking about it. ACE is angiotensin converting enzyme. When we talk about the ROS system, that means renin-angiotensin-aldosterone system, the angiotensinogen is converted into angiotensin 1, and that is converted into angiotensin 2 with the help of the angiotensin converting enzyme, which is secreted from the lung. Whereas actually the ACE2 is actually the monodecarboxylase, peptide decarboxylase. It removes actually one of the amino acid from that particular aspect. So in that particular case, there is a slight difference. We will talk about that particular thing. Transmission, when we talk about it, it is a human-to-human transmission. Previously, it is only thought about the droplets. And when we talk or sneeze through the nose or actually the throat, the droplets move. That is one reason is the social distancing when we are talking about it. But the most important aspect is now people are concentrating on aerosol formation. Because when the aerosol, when we talk about it, the distance it can travel up to 6 meters, even more than 6 meters, 28 feet, it can go into the particles are suspended depending upon the ventilation is one of the most important aspect, and the direction of the air conditioner flow, because in the summers people use actually the air conditioners. So in that particular case, in the path of the air conditioners, people sit and the drop in the aerosol formation is moved for a longer actually the distance. That is one of the most important aspects which we have to understand. This aspect I am not going to talk already in detailed pro-inflammatory. Whenever we talk about immunopathology, when we look into it, this actually the decrease in the count of or inactivation of the T cells is reported, and pro-inflammatory, and the whole question comes a series of actually the cytokines which are actually the changed. But the previous speaker has talked about kinetics, one of the most important aspect. As the disease progresses, this particular markers do vary. In that case, the kinetics of this particular parameters need to be followed. Yes, certainly it is a difficult parameter to be determined. That is kinetics, what I'm talking about in normal healthcare centers. So in that particular case, when we look into it, the leukopenia, he talked about it, lymphopenia, thrombocytopenia, and elevated CR level, CR protein levels, and of course reduced CD and CD8, CD4 and CD8 T cells in severe infection. The complications, it actually reaches into not only the lungs, it is acute cardiac injury is reported, acute kidney injury is reported. One of the most important thing is diabetes uncontrolled, it goes into CKD, unfortunately. So in that particular case, one of the aspects which we have to understand, and the secondary infection and the shock and pancreatitis is another thing. The recent reports, I think day before yesterday, is indicating that in organ transplanted, this is actually the severe, because organ transplantation means people are using actually immunosuppressants. So in that particular case, disease spreading and fatality rates are very high. So in that case, antibody development is very less in these actually immunosuppressant receiving the patients. This is where actually I want a little bit spend the time, which is a little bit different in this particular case. This actually, the virus is having the spike. It is all actually the structure what we have talked about it, and this actually receptor binding domain interacts with ACE2. Which is actually present on the cell membrane. It is actually the case, and the RBD actually combines. RBD means a receptor binding domain. It is also having the catalytic site. Catalytical site is converting actually the case, and the question comes here is angiotensin 2 acts on ARB. That means angiotensin receptor blockers acts on this thing, block this response, and which causes actually the vasoconstriction, because direct stimulation leads to vasoconstriction, rise in blood pressure, that is angiotensin, and fibrosis, inflammation, and decrease in actually S2 expression. This is one of the most important aspect. Whereas actually the other catalytical activity of S2 is producing angiotensin 1 to 7. That is one of the important aspect. This particular actually the angiotensin 1 to 7 act on MAS receptors, and it causes vasodilatation and actually cardiac production, anti-inflammatory and anti-proliferation, anti-thrombosis. So in that particular case, there is a balance between the AS1 or AS, we'll talk about it, or AS2, one aspect, when it is playing role. One causes actually the increase in the vasoconstriction, another causes actually the vasodilatation. With this particular concept, people said whether actually the angiotensin receptor blockers need to be administered. This is one of the biggest controversy, but later it was actually found that it is people, all actually hypertension associations, they talked about it. You have to continue with actually, as in case of the patients who are having actually the diabetes and hypertension. Now, when it actually, after combining with it, this particular actually, this is actually the attachment and the TMPRSS, and it's actually the cleaving site into S1 attachment, and once actually this cleaving takes places, the fusion of actually the virus will take place, and there actually it endocytosis mechanism, it is actually the internalized. This is the second step. The third step is proteolysis. This goes into endosome. Now within that endosome is actually the virus, but we have to actually take out this particular uncoating of the virus. Single-stranded RNA must takes place. So in that particular case, this actually the replication part takes places, and you have actually RNA transcription and translation taking place, where this actually the activity takes into picture, and then actually the translation and the structural actually accessory proteins, they are assembled in the Golgi body, and the new virus, mature new virus. That means the virus which has actually entered in the body, it is actually replicates in number, multiplies in several numbers, depending upon actually the immune system, immunity of the host, that means a human being, and the spread actually takes places, and the disease progression takes places. So this is actually by exocytosis, it releases and it goes into other actually the organs and start actually the infecting. So this is the major point when we are talking about it. Now, at the various stages actually, and the drugs do act. Chemostat is actually the inhibiting this particular interaction, that is actually the inhibiting TMPRSS2. So with that particular thing, the virus do not actually binds to it. Now the next aspect is once actually the fusion actually takes place, comes into sight, the process needs to be converted, and it comes out in the single stranded. There actually hydroxychloroquine acts. this is the most controversial. Unfortunately, it has politicized, and the pharmacology of this particular thing is a very important. Indian actually ICMR recommends as a prophylactic, so that it can actually prevent the further steps if it is started actually the taking it. And the people have used actually after this particular thing, severe conditions, along with the and it started actually reporting several actually the deaths because of the drug interaction, and QT wave prolongation is one of the major actually things that occurred, and people actually, once it is approved also, emergency approval for this purpose, it has withdrawn. The other actually the point, lopinavir and other antiviral drugs act on this particular process. Here on the M protein are actually 3CL4 the aspects, and the RNA dependent RNA polymerase. When we talk about it, remdesivir, which is now approved, in that particular case, people are using this particular thing, and it can actually prevent it. The another aspect what we are talking about is how actually one is actually the treating the virus and actually the interfering with the virus, actually the mechanisms, and another aspect is the symptoms. When it is actually coming into the lungs affected into the case, and it is neutrophils and cytokine storm, as it is already mentioned about it, previous speaker detailed, and IL-6 and TNF alpha is also important role it actually the place in it. There it is tocilizumab was initially the monoclonal antibodies were successful, but in the severe conditions, as it is, I fully agree with that particular concept, which is actually the case. Now it is not actually recommended in the serious cases. So this is actually, there are no specific drug for actually the what you call SARS-CoV-2. That is one of the most important aspect. So in that particular case, new drug development for this particular virus, it is a long-drawn process. Drug discovery of bringing a new actually the drug, it is actually takes somewhere around previous actually the protocols wise, it is 15 years plus. So in that particular case, existing drugs, people actually used it and finding out whether actually it is useful in that particular case. Because these are actually some of the monoclonal antibodies, JAK-STAT inhibitors. And when we talk about lopinavir and ritonavir, we are actually, again, antiviral drugs. Flavipavir, which is other infections, which is approved in certain countries. Remdesivir is approved. Yes, it is actually RNA-dependent RNA polymerase acting on this particular thing. Recently, Indian actually the company, which is actually the Zydus Cadila, Virafin, signaling proteins, pegylated interferon alpha 2b. Is actually the approved by the DCGI. When we talk about actually the drugs which are actually repurposed drugs for immune modulating therapies, when these are actually the when we talk about this particular case, disease modifying, conventional synthetic disease modifying, first category. This is hydroxychloroquine is having the effect, but somehow everyone actually criticized this particular thing. Glucocorticoids, especially these are found to be very effective. In that case, series of structural analogs which are in this thing, and in certain cases actually other actually the substances which are producing, and biological actually the aspect. Initially, this particular thing is not actually now recommended. Anakirana and IL-1 actually receptor aspects, and targeted ones. Baricitinib and actually other actually the TNFs, which are actually the targeted actually disease modifying substances which inhibit this particular thing. And stem cells also people have started. This is morning also we talked about it, the first speaker, And the question came on that particular thing in the new variants. It is still actually this needs to be proved in this information. This is one of the treatment, MAC plus the treatment, what is actually talked about intravenous methylprednisolone and plus high dose of ascorbic acid in the severe actually the cases. Thiamine is playing role, vitamin B1, in improving the status, low molecular weight heparin, and plus actually, plus means other actually the statin, zinc, vitamin D, and famotidine, which is actually acidity to decrease it, magnesium, and melatonin. See, when we talk about incubation period and actually symptomatic, viral actually replication is taking place, and early pulmonary phase and actually the late pulmonary phase where immune dysfunction. And see, macrophages start activating. In that particular case, they start causing harm to internal cells, body cells, save its own cells. That is one of the major actually the problem which actually comes into the picture. And these are the treatment profiles when we talk about it. Now, let me actually go into vaccination, which is the fundamental point. There are two actually the important aspects which we have to talk about it. Mucosal and the systemic immune responses to the natural infection and to vaccination. This is one of the most important aspects when we talk about it. The first figure when we talk about it, natural infection. That means whenever the first actually the case occurs and the virus enters in the body through nasopharyngeal route, it actually infects it. At that time, the mostly soluble IgA1 is actually present in the upper respiratory tract. That is one of the most important aspects. Whereas in the lungs, we do have actually the mostly IgG1. This is one of the observations reported by Kramer in this particular case. Now, when you administer actually the vaccine, either by intramuscular or actually the intradermal, this is how actually the some vaccines can be intradermal, but mainly the intramuscular aspect. They actually mostly develop actually IgG1 in the lungs, not actually upper respiratory tract. This is one of the most important aspects which we have to understand. So in that particular case, even after vaccination, if an infection is come or any particular aspect, upper respiratory tract cough is going to persist. This is one of the point which we have to understand. So what is actually the answer in that particular case? People are trying for actually the newer method of actually the vaccination, that is intranasal vaccination, which is planned to administer this particular thing via actually the nasopharyngeal route. And nasopharyngeal route, when you administer actually a vaccination, it is going to actually provide the soluble IgA1 in the upper respiratory tract, but the amount of IgG1 is actually less in the lungs. So this is one of the aspect what actually the recent literature talking about it. So with that background, these are actually the normal, whenever previously vaccines, people started actually asking, it used to take 15 years long procedure, the design and exploratory and processing and preclinical toxicology, and then IND submission, phase 1. It is a sequential, phase 1, phase 2, phase 3 are sequential. It takes actually a long time, 2 years plus 2 years, 4 years, 4 years plus 3 years, is 7 years, and then actually the biological license approval authorization is submitted, they take actually 1 to 2 years, and large scale production 15 years long. It is a conventional traditional development. So in that particular case, in a pandemic situation, they have actually the pandemic approval procedure. Rapid approval procedure is actually followed. That's why it is emergency utilization authorizations are given. So in this particular case, when we talk, what is the difference in between is when we are talking about the simultaneously these clinical trials are actually taken place. That is one of the major aspect. So with that particular thing, with 1.10 months to 1.5 years, one can actually come out with this particular thing. Now, what is the vaccine aspect? So one is actually the spike protein. People can use it, actually the RBD of the protein spike. Inactivated vaccines, in this particular case, by chemical method, the vaccine is completely inactivated. It is intact, and every actually protein which is present is administered. We'll see the examples. And live attenuated vaccine is another actually the way of this thing. And the third one is recombinant spike protein based vaccines and recombinant RBD based vaccines. So that means receptor binding. This portion is the receptor binding in your spike protein. So this actually, then next one is vaccine like actually particles carrying, and this is our nanoparticles which are developed. They actually have these spike proteins on it in their structure, but they do not have the genetic material, genome, and no genome actually they present. Other actually the methodologies, replication incompetent vector vaccines, which are actually they present in the case, and the replication competent actually the vectors, and inactivated the virus vector, which can be inactivated in this particular case, can be used, and DNA vaccine consisting of actually plasmids, and DNA vaccines consisting of RNA encoded spike protein, which can be used in this particular case. Now, what are that particular actually based upon these particular technologies or platforms? Certain actually the vaccines which have been approved, which are presented in the next few slides. Bharat Biotech, which is actually Covaxin, which we talk about it. It is an inactivated virus, and SARS-CoV-2 is chemically inactivated using actually the beta propiolactone. So it cannot actually replicate, but all the proteins remain intact. It is originally, when it is a clinical trials, when we talked about it, unfortunately, when on January 15th, they have given for this particular vaccine what you call clinical trial mode. This created a confusion in a lot of actually the people. Now the data is available with the phase 3 studies which have been conducted. It is in this particular case, it is a whole virus. This particular COVAX is the whole virus is represented to immune system, presented to the immune system. It is inactivated, and this immune actually response is likely to target not only the spike protein of the this thing, but also matrix and actually the envelope and nucleoprotein. This is that is people at that time when this slide is prepared, they have not talked about other variants efficacy. Recently, ICMR news, I have not seen any scientific paper. That is one of the most important aspects. But they are claiming that COVAX is having actually the activity against the variants which are actually available, double variant when we are talking about it, not triple variant information is not there. The second variant, the second vaccine which is available is Oxford AstraZeneca. This is actually the viral vector vaccine, which is actually double-stranded DNA encoded for the spike protein, and it is actually administered, and this particular thing is Oxford University and AstraZeneca based actually research, and they have given the license to produce to Serum Institute of India. It is available. Initially, it is talked about actually the second dose need to be administered after 4 weeks of the first dose, but now they have increased being the activity the most important aspect in the clinical trials, whatever the reports says, viral replication in the upper respiratory tract was not controlled. This is one of the point about the Covishield. This is the reports which are available. And the third one, this is actually Gamaleya Research Institute, Russian actually the virus vaccine, and this particular Sputnik, it is V, not Roman figure 5. This is one of the most important aspect. It is actually the adenovirus based actually DNA viral vector vaccine, and this is recently approved in India, and it is arriving. It is first manufactured doses are coming from actually the Russia on within next 2 days. That is what the information is available, and it is actually the effect. Originally they talk about 91% because it expresses the spike protein on actually the human cell because of this particular vaccine which is actually coming into the picture. This is actually the Moderna, and the next one, when we talk about the Pfizer, more or less they are same, but they are two different organizations from the United States. So simultaneously they have come out. The encapsulated mRNA vaccines. This is mRNA vaccine, which is first time used in actually the developing vaccine for any particular disease. Though this technology people are actually platform is being tried for the last 10 years, no approvals have come or no success story, but anyway, this is actually the success story, and high efficacy is actually shown in this particular case. In this particular thing, and the messenger RNA is actually in the human cell is on the body now expresses actually the spike protein, and that the immune responses will actually develop in this particular case. This is the same in this particular case technology. This is another company, the Pfizer and the United States. They are working actually together with Germany, Bio and Tech, German and actually the United States collaboration with that particular thing. This is actually the developed aspect. Okay. The next one is the Johnson & Johnson. Is approved in the United States. Again, it's stopped for some time, but again restarted in this particular case, viral vector vaccine, which is actually adenovirus 26. From there, actually, it talks about this proteins which are actually developed, and this actually Johnson & Johnson is actually the active against the UK South African strain. That is one of the things they have already, by the time this is clinical trials were able to do it in the case, and they have reported this particular activity. Novavax. This is Novavax is a Nova Incorporation's United States product. It is a virus-like actually particle vaccine. This particular thing which is approved in this particular case, and efficacy is also shown to UK variant B117. It is showing efficacy in this particular case right at this moment in virus-like particle, which is actually. Of course, China is also having actually the case, and China exported this particular thing to Brazil, and still there are actually, this is also an inactivated virus. Whatever the reasons, Brazil has taken an understanding and purchased actually the thing from actually the China, and results are not encouraging what actually the people talk about it. Now, when you talk about the comparison of this particular thing, when I'm talking about formulation aspect and especially storage conditions, because in now the temperatures are already reaching 30 degrees centigrade in this particular aspect, maintenance of this particular thing, stability, you have to maintain at -20, the Moderna vaccine, and whereas actually the Pfizer, it is -70 degrees. In this particular case, at the refrigeration, it can only stay for 5 days. That is one of the aspects which is actually coming into the picture. In the case Johnson and Johnson, it is for a long period. It can actually, one of the most important about Johnson and Johnson is a single dose. In this particular case, is sufficient enough to produce the vaccine. So this is all about, actually, I can talk about morning already, the different strains talked about in detail, which is actually in the tabular form. What are the changes, key, actually the mutations which are there, what are the points which are explained. I am not going to detail because Dr. Varma has explained all these particular aspects. This is actually the double strain mutation, V1617 is also. Some data which actually the people talk about it, infectivity is much faster. The mutation which is actually taking place at actually the R is converted into L452, it evades actually the antibodies. This is actually one of the most important aspects. So that's why immune evasiveness is coming into the picture as per the reports. The second change is also, and the Q is actually changed to EQ 484. So in that particular case, it is actually immune, actually the evasion, and better fit actually the binding of the protein. In that particular case, this particular actually the strain is actually reporting. We have actually what you call the Indian SARS-CoV-2 consortium of genetics, and that is INSACOG. This is present actually in India on the sudden search. What they are talking about it, as per actually the data, it is yesterday's data is 3.6 lakhs daily. It is changing in this particular case. In Maharashtra, in the first time it is reported, now 6 more states have been, 10 more states, so total altogether until April, actually mid-April. It is not only in India, as it is mentioned, in 11 other countries also reported this particular strain because of the travel, as in the morning was actually discussed about it. Now, what is it actually, the vaccine production capacity of Indian capacity? That's what we have to understand about it. Prior to COVID, India is actually producing 60% of world's vaccines. That is different other diseases, whether it is hepatitis C, whether it is hepatitis B, and other actually the vaccines, we are producing 60% of the world's vaccine population. Now, COVID point of view, 3.5 million doses. We are second to United States. They are preparing around 4 billion. We are in this particular case. So the Covishield capacity is 70 to 100 million doses every month, and COVAX is 12.5 million a month. This is one of the major aspects. Unfortunately, the what you call the vaccine nationalism is actually taking place. This particular point, US actually has implemented a Defense Production Act. With that, actually, the raw materials which are required for the production is actually prevented by export of these things. That is one of the major, actually, the constraint. Recently, that particular thing, they have removed it after the repeated requests. India, when we are talking about it, Serum Institute of India, Bharat Biotech is also working on nasal administration, nasal vaccine, and you have the Panacea Biotech, Sanofi Santa Biotech, and Biological Events, and Hester Biological Sciences, Jidus Cadila. Right at this moment, availability when we talk about Serum Institute's Covishield and Bharat Biotech Covaxin, and the third one is approved in India is Sputnik V, which is actually in collaboration with Dr. Reddy's laboratories. The clinical trial phase 3 are being conducted, and capacity of the 8.2 billion doses per year when we talk about it. India followed a policy till recently to research, that is vaccine Maitri. In this particular case, vaccine started on 16th January, 3.5 months back, somewhere around 104th day, if I'm correct, of the vaccination. This particular thing, we have actually donated or exported and corona vaccines to the neighboring countries as on actually 13th March, somewhere around 66 million. These are our neighboring countries for which we have done and supplied it, and these are in the pipeline which we are going to supply for this particular country. What is the take-home message in this particular case? SARS-CoV-2 is a new coronavirus, highly contagious, transmitting via the droplet and aerosol formation, affecting multiple organs. So death rate, forget about it. That varies actually. No specific treatment or drug for actually the treatment of COVID-19 antiviral. When we are talking about it, new drug discovery is a long process. Existing drugs are repurposed, and which is less time consuming and cost effective. Several platform technologies are being actually used to develop the vaccines. There are, at this point, I have not, I think, mentioned that somewhere around 182 vaccines are in clinical trials already throughout the world. When we talk about clinical trials, the site, the mRNA-based vaccines are first time used to develop corona vaccine more correctly, and some of the vaccines are given actually in the emergency use authorization by the drug regulatory authorities throughout the world. Due to digital technologies and information and data is plenty of available, at the same time fake news are also circulating. That is one of the most important aspects what we have to understand. When the mind is delusioned by the fake news and the negativity, your inner conscious should prevail for transparency and unbiased to achieve equity, diversity, and inclusion. Non-pharmacological interventions such as mask, people are now recommending double mask also. The social distancing, avoid large gatherings, cross-ventilator space, and washing his hands either by sanitizers and aspects. And thank you very much when we are talking about it. Okay.
Thank you, Professor Varma. A wonderful lecture. Where do we stand on the development of antiviral drugs?
As I have already mentioned, one of the most important aspects is new drug development will take a long time. This is one of the drawbacks because the processes and everything, people are now working the newer technologies, artificial intelligence, in actually the identifying what type of the drugs which are actually acting on the virus and finding out this particular aspect. hopefully the new antiviral drugs will come out. Because remdesivir, as it is mentioned, it is Ebola virus. And because it is in a short time, it has come into picture. And same way, we have to wait for it, and I cannot give any exact this thing. But question comes here is the human beings should develop their immunity. That is one of the major aspects which we have to look into it. Otherwise, it is going to be difficult.
Follow-up of that question would be, the market is flooded with a lot of these compounds that say that they are, immune boosters. How much, as you said in your final slide, when the mind is overcome disillusioned with fake news and negativity, what, as someone who has spent a lifetime in pharmacology, what do you suggest when it comes to immune boosters, and how much of it is relevant, and what if key elements, say for example 3 or 4 drugs or substances that you can suggest that could be for sure immune boosters have been shown in multiple studies, rather than going by WhatsApp forwards? Can you suggest a few?
It's very difficult, sir. It is one of the most important, as you mentioned. Whatever we are actually getting in WhatsApp University is one of the most obnoxious information which we cannot rely on it. People will talk about, is it a pseudoscience? Yes, on the other hand, China, actually, the traditional medicine is successful that they are claiming. I do not know what is the reliability of that particular information. They have spent a lot of actually the research, money and aspect, time and infrastructure in actually developing traditional medicine. We require a lot of actually the research in validating these claims. This is one of the most important aspects. To some of the things they have claimed, they have given actually WHO approvals. Then afterwards, WHO came out with a notification stating that we have not given any approval for these products. I do not want to name the company, but the question is, you can understand what I am trying to say.
The floor is open for questions by the audience members if they want to ask any question. Professor Ramarao is a very experienced person. You can ask even layperson questions. Don't be shy in asking. Dr. Shiva, if you have any questions, you can unmute and ask.
Yes, good. I think Shiva is good. Men should get the vaccine after recovery, not for me.
Sorry. Sir, I have one question. Like regarding emergency use authorization of vaccines, like which component you are thinking very important, like whether efficacy or safety? I don't expect both. If you want to prefer one thing, whether efficacy or safety, which one we have to consider?
Efficacy, they have decided 50% is the minimum requirement. Okay. That is one of the major aspects which we have to understand. So anything about 50% as per the WHO guidelines, I'm talking about it. This actually varies from country to country. That is even Swaminathan, madam, is working at actually the WHO, and they have actually given this particular direction, one important aspect. Okay, safety is the most important aspect, but nothing is actually free from toxicity. Paracelsus, we talk about 16th century. It is the concentration which makes any substance safe. You cannot classify any substance as a safe or actually this thing. It is only the dose which makes actually the case. All vaccines are reported, including mRNA-based vaccines. Thrombocytopenia has been reported. 2 cases have been come out with actually Moderna vaccine as per the report. So in that particular case, because antiplatelet factor 4 is actually playing role in this particular thing, people have come out with actually this particular aspect. So Europe is it again a pharmacogenomics? I do not know. Why only Europe is reporting 39 cases of actually thrombocytopenia and the blood clot formations, dysfunction of this thing, and the thrombocytopenia is coming into the picture. Whether actually other places also there, they are not testing, are not reporting, we do not know that particular aspect. Unless this is a new disease, as you are fully aware of it, information is evolving from all corners of the world. And during that process, as I showed, infodemic, number of papers, everyone, even actually the people are questioning the editorial policies have been relaxed in publishing the papers. So that is what people actually talking about it. And now plenty of that particular type of questioning will come on the papers which are published. That is one of the aspect which you have to say. That's why it is very difficult to give a verdict or actually a statement on this particular aspect. So yes, someone actually asked the questions after actually I was reading actually the points. After getting infection, is it necessary to vaccinate? Yes, as per Fauci, the authority on actually this particular aspect, it needs to be vaccinated. The persons who need to be vaccinated. What vaccine, whatever it is available, we have to do it. That is another question which we have to say. And people are talking about Covaxin is better. That is again a WhatsApp information. I'm not going to certify it because these are the few points.
Sir, for treating the patients of mild to major COVID infection, can we use the monoclonal antibody instead of the remdesivir?
It is a very cost effectiveness also we have to think about it. Any monoclonal antibody is costs a humongous task. This is one of the aspects which we have to understand. Ultimately, where actually the general population which are there, it is not an easy solution. Yes, highly affluent people can work out that particular aspect, in my opinion. And the cost factor of the biological, actually the solutions, it is not an easy task. Monoclonal antibodies.
And also second option, sir, like remdesivir having a very costly, and can we treat it, replace it with a steroid, which is cheap as compared to?
I have shown actually this particular point, probably it is missed. Steroids should not be used up to one point because it is immunosuppressants. The body immune system should work up to some time. Then only when it is in the severe, the time of actually, that's why I said phase 1, phase 2, phase 3, phase 4. It is only the steroids is an alternative, or actually the treatment should be started after actually incubation, after actually the replication part. Then only the steroids needs to be implemented. Otherwise, you are suppressing the immune system. What good it is? Ultimately, the human body has to actually respond. When actually the antigens are entering into it, you have to allow it. On the first day itself, if you start actually steroids as a prophylactic or actually a wrong treatment, it creates havoc, in my opinion. This is my opinion. I have not got any data, but people have recommended MAC plus treatment profile also says corticosteroids, as a methylprednisolone is starting after 7 days, 7-day treatment. It is not initial treatment. It cannot be. It should not be.
Okay, thank you.
Okay, thank you, Professor Ramarao, for a wonderful lecture. Now I hand it over to Dr. Monisha.
Good afternoon. Thank you, sir. Thank you, all the speakers. So I am here for a few closing remarks and vote of thanks. So thank you, all the speakers. In the morning, we started with Dr. Subhash's lecture, where he talked about role of genome surveillance why, whether it's necessary. Yes, the way he presented the work. Yes, the various. It is important to identify various variants of interest and what is the cause of reinfection. Amazing lecture by Professor Dr. Subhash, and I'm sure all of us have learned, all the participants, and they have learned a lot from these presentations. So from Dr. Subhash's lecture, the take-home message was that irrespective of whether you are getting reinfection or whatever, get vaccinated. That is what was his take-home message. Then we have Dr. Shiva. Thank you, Dr. Shiva. That was an amazing lecture. Your presentation was too good. And as a clinical physician, I understand we don't go to that level, and your expectations from our researchers is there. So that is what the way you started about the cytokine storms and various indicators and role of cytokines, IL-1 beta. Earlier, when you started, I was surprised when you said that previous report said that IL-1 beta is not that crucial. But then you said, okay, recent reports are there that IL-1 beta is again important. But yes, IL-6, that's one crucial, TNF and IL-6. The role of cytokines is very important. And then you talked about the earlier indicators. And from your lecture, what we could understand is that as a physician, we really want to collaborate. We really want to work in hand so that we can find out some early. Our concern is that the patient should go. Your concern is that we should identify things early, right?
Yes,
So it would be very great if we can identify some early indicators or early markers. We can distinguish the mild symptoms versus severe versus very chronic symptoms. So definitely your take home, from your presentation, the take home message was that yes, clinically, your physician wants, there should be some markers, peculiar markers where the patients can be identified, their severity of the disease can be identified at a very early stage.
Yes.
Thank you, Dr. Shiva.
Thank you.
Then we have Professor Rama Rao. Thank you, sir. Thank you for your amazing talk. You started with the comorbidity, that is your expertise. Diabetes and hypertension is your expertise, and it's an inspiration when we see your book review which you published last year and the kind of work you have done in that review. So that was really amazing. And then the angiotensin blockers, you started with that, the patients with comorbidities, you talked about that. And then you talked about various, that's really, I'm sure that including me and all the participants, it was amazing when you compared the drugs, their efficacy, their mechanism of action with that pictorial presentation. That was amazing how various drugs, they work during the viral infection, and the comparison between various vaccine, their efficacy, their formulation, their storage, regulation and policies, everything was, those things were really amazing, because some of our students, they have, and even us, we always have these questions and queries. So I think it was a very nice presentation. Thank you very much. And take home message from your talk was that, okay, we need to avoid fake news. We need to be positive, stay positive, and rely on the authentic news, authentic scientific data. Thank you very much, sir. So last but not the least, I would like to thank Honorable Vice Chancellor, sir, who has agreed and who has been there all the time. And Professor Husrika was there in the morning, so he's there, DIA, sir. And all the faculty of our department, we have been working for this event. So, and the computer center staff and all the people who have worked and all the participants, thank you very much. So before we end, I would like to add that we'll be joining again at 1:50. So there would be another session where Professor V.S. Chauhan from director of ICGEB, and he was also chairman UGC. He'll be joining us, renowned immunologist. He'll be joining us at 2:00. So you all are invited again to join the same link and join us. And there will be some student presentations. Very nice student presentations would be there about the frugal innovations during pandemic and some awareness videos. So you are most welcome to join again at 1:50. And thank you very much. Thank you all. Thank you. And now please rise for national anthem. Thank you all. Thank you very much.
Punjab, Sindh, Gujarat, Marathwada, Dravid, Uttara, शुभ नामे जागे, तब शुभ आशीष मांगे, गाहे तब जय गाथा, जय जय मंगलदायक, जय हे भारत भाग्य विधाता, जय हे, जय हे, जय हे, जय 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 हे.
Thank you. Thank you all. Sukhna ji.